Good morning, everyone. Yes, Sumit. Shh. Thank you all for coming to day 3. Hopefully, you had a fantastic 2 days. We have another full day today, so hope you're rested. I've been told there's plenty of caffeine, so get yourself caffeinated or whatever your poison of choice is to get you through the day. So our day 3 is focused on applications of PBBM. In the first 2 days, you heard about the in vitro inputs, model development, validation. So today we're going to focus on application. The flow of the day is going to be identical to the first 2 days. We have a morning full of speakers and in the afternoon, 4 breakout sessions. So with that, I will let, and by the way, my co-moderator here is Bhagwant Rege from FDA. And Bhagwant, so introduce the first speaker, if you may. Thanks.
Sure. So we are going to have presentations from industry and regulators alternatively. So the first speaker is Shivacharan Kolipara from Dr. Reddy's Laboratories, and he's going to discuss applications of PBBM in generic product development. And he's not here with us physically, but he's going to give a virtual presentation live.
Hi, am I clearly visible and audible?
Yes, you are. Go ahead.
All right. Can I start?
Yes, please.
All right. So good morning, everyone. Thank you so much for giving this opportunity to present in this wonderful workshop. And as Amitabh was mentioning a couple of minutes back, I hope all of you had a great time with respect to learning different aspects of PBBM modeling and its applications. Today, I'm going to talk about the application of PBBM modeling in the generic product development. And we are going to discuss a couple of case studies as well. So before I start with the discussion, let me tell you that the opinions expressed in this particular presentations are based on my view and do not represent the opinions of Dr. Reddy's Laboratories. So this is going to be the short agenda for today's meeting. We are going to see how biopharmaceutics or PBBM modeling is going to drive the generic product development, and what is the modeling framework from the generic perspective, what is the importance of resolution, and how do we see biopredictive versus QC media resolutions. And we are going to see a couple of case studies in terms of critical valuability attributes evaluation, and how do we utilize PBBM modeling for understanding the impact of faster resolutions on safety. Then something about utilizing PBBM modeling for evaluation of the gender impact, and final case studies with respect to utilization of DDD plus for evaluating the discriminatory power of the resolution method. And we will focus on some of the common regulatory queries on PBBM modeling, followed by the way forward. So by this time, you would have seen various presenters talking about the applications of PBBM modeling throughout the life cycle of the drug product. But just to recap, at the preclinical development, PBBM modeling is used to understand the formulation selection for the toxicological studies. And as the molecule or the product moves to phase 1 or phase 2, then PBBM modeling can be utilized to evaluate the predictions or exposures of the first-in-human formulation, and to understand the impact of various physiological parameters. And then once we get the data from phase 1, the model can be further defined, and the model can be applied at phase 3 and phase 4 to support the formulation development, which is going to hit the market. And at this point of time, we are going to use the model for setting up the patient-centric dissolution testing, and to use the model for risk assessment with respect to formulation changes or the site transfers. And once a molecule is on the verge of launch, or once it gets launched, then the focus shift towards the generic medicine, which we are going to talk in detail in today's presentation. At this particular stage, the modeling can be utilized to understand the bioequivalence risk assessment, and to do the BE projections, and to understand the impact of different formulation variables on the bioequivalence of the generic product. So this slide, or this can be called as a master slide that talks about various applications of PBBM modeling in the generic product development. And typical applications where PBBM modeling can be utilized is to justify the resolution dissimilarity or the F2 mismatch, and to establish the clinically relevant dissolution specifications or dissolution safe space. And wherever there is a F2 mismatch and when there is an issue for biowaiver of the lower strength, it can be utilized. And most importantly, there can be differentiated formulation that the generic industry develops in terms of fixed dose combinations and other things. And PBBK and PBBM modeling also can be utilized to support the drug-drug interaction study waivers in those cases. So these are the famous regulatory guidances which describes the utility of the PBPK and PBBM modeling for new drug applications as well as the generic applications. All of you present in this particular meeting are aware that US FDA guidance that has become effective in October 2020 talks about PBBM modeling in terms of supporting the manufacturing site changes and process controls in order to support the quality risk assessment in the pharmaceutical industry. EMA guidance talks about PBBK modeling utilization to support new drug development as well as the generic product development. And then we also have this particular guidance from WHO. And what is interesting to see is that apart from the agencies like US FDA, EMA, other regulatory agencies such as Brazil and then New Zealand, China, they're also open to PBBM submissions. And we as a generic industry, we had submitted PBBM-based justification to a few of these countries. And then we have received some encouraging response on these models that we have submitted. So this is the common workflow or the methodology for the development of PBBM modeling. I think, again, all of you are aware. But what is important to note is that few aspects from the generic product development or from the generic industry. So in terms of the model setup, we typically gather the model parameters from physicochemical properties or the disposition kinetics. And then we incorporate the data of the dissolution through the standard dissolution models that are available. But as a generic company, it may not be possible that we always have the input parameters data for all of the parameters, especially the transporter kinetics and the enzyme kinetics. In those cases, we might need to do some kind of an optimization. But then when we are using these kind of models for regulatory justifications, we need to provide the adequate justification for the input parameters. Then in terms of the model validation, typically the validation can take place against the literature data. And then with the in-house data, especially when we have huge amount of individual plasma concentration time profile, the validation can take place. At this point of time, we can also use parametric sensitivity analysis to understand the rate limiting step. And once the virtual population has been developed, then we can go to the third step in terms of model application, which can be B projections or dissolution safe space or justifying the dissolution specifications and so on and so forth. So talking about the dissolution, this is a single chart that demonstrates different options that are available for integration of the dissolution data into PBBM models, especially from Gastoplus perspective. For immediate release formulations, we can use direct dissolution as an input, and we can also include the particle size as an input, which will take care of the in vivo dissolution. And within the dissolution inputs, we can have single Z factor or multiple Z factor. And especially when we are using Z factor or multiple Z factor, we need to understand the limitation that comes with Z factor with the adequate justification. When it comes to modified released formulations, again, direct dissolution input can be used, wherein we are assuming that in vivo dissolution is equivalent to in vitro dissolution. And we can also use some time scaling approaches, and then we will fitting to fit this dissolution data into the models. When it comes to the delayed release formulations, they act as immediate release formulation after they pass through the gastric phase. In those cases as well, we can use Z factor as well as direct dissolution data input with the limitations of Z factor as we have discussed above for the IF formulations. So how do we utilize this particular dissolution data input into the modeling in terms of establishing the bioequivalence safe space? So what you see from this slide is a demonstration of bioequivalence safe space for the innovator drugs as well as for the generic drugs. Although the methodology of bioequivalence safe space establishment remains same between innovator and generic drugs. But in case of innovator drug, the pivotal phase 3 formulation or to be marketed formulation is taken as a reference. And then we can have different dissolution profiles surrounding that. Whereas in case of generic formulation, we have reference formulation as well as a test formulation. And typically we consider pivotal test formulation as the reference formulation surrounding which we will have a couple of dissolution profiles. So once we input these dissolution profiles, either coming from direct laboratory experiments or from virtually generated dissolution profiles, we can input these dissolution profiles into the modeling, and then we can try to establish the safe space. So once the safe space is established, then we can utilize the safe space to understand the impact of critical bioavailability attributes, which we will discuss in the first case study. So these attributes can be process variables or the material attributes or the formulation variables. And then once you define the safe space, they also will be helpful to understand the impact of critical bioavailability attributes, through which we can also use PBBM modeling for the clinical quality along with the manufacturing quality. So on this particular slide, we try to summarize our learnings in terms of OGD or the QC media versus the biopredictive dissolution media. So what we have done in this particular case is, as a generic company, we generally develop or we are interested in development of a biopredictive media for the bioequivalence risk assessment, as well as a couple of other applications. So we have a lot of products, and wherein for each product, we have gathered the data of biopredictive media. And then we try to correlate the conditions of the biopredictive media with that of the conditions that are defined in Office of Generic Drugs, which is typically used as a QC media. So what you can see from this particular summary for pH, the RPM, and the volumes is that there are distinct differences exist with respect to all of these conditions when we compare biopredictive media with that of the QC media. So typically higher pH conditions are used in the QC media, and again, typically higher RPM coupled with higher volumes are used in the QC media as compared to biopredictive media. And generally, in case of biopredictive media, we work with lower volumes and then lower RPM in order to get the discrimination. But what this is telling as an overall summary is that there are distinct differences exist with respect to QC and biopredictive media. And although the QC media can take care of the manufacturing quality, but we need to have a separate media which can take care of the biopredictability, so that you can have both manufacturing quality as well as the clinical quality in place. So to further elaborate this particular concept, on this slide, we have talked about QC versus biopredictive media and the corresponding risk assessment. So overall, there can be 4 different scenarios. So the first scenario is, which is also called as the best case, where if the QC media is biopredictive, then this represents the best case, and only one method can serve both purposes, the quality from the manufacturing as well as the quality from the clinical. But in case when the QC media is not biopredictive, then it represents a medium risk, and then we need to have 2 different methods, one for manufacturing quality and another method for biopredictability. And then the third case, where the QC media is not biopredictive, and then we have different biopredictive medias, one for fasting and one for fed condition, then obviously we would be requiring one QC method along with 2 biopredictive method. There are also cases where we are unable to identify any biopredictive method, and then it becomes challenging to answer the regulatory questions, and we need to rely more on the biopharmaceutical risk assessment rather than focusing on the biopredictability of the media. So these are some of the cases which generic company faces regularly, and as most of the queries that are received on the QC media with respect to in vivo discrimination, we need to consider this particular chart appropriately, and we need to have biopredictive medias developed appropriately. So with this background, we will move on to the case studies. The first case study talks about the evaluation of the critical bioavailability attribute. And critical bioavailability attribute can be defined as a formulation or a process variable that is expected to critically impact the bioavailability of the drug product, and it can be a material attribute or a formulation variable or a process parameter. So as a generic company, recent trend, we see that most of the regulatory agencies are asking us to connect the traditional formulation risk assessment with that of the in vivo quality. Traditionally, the formulation risk assessment is based on in vitro evaluation or in vitro impact of these variables, and extending this particular in vitro evaluation to in vivo evaluation with the help of tools like physiologically based biopharmaceutical modeling can help us to embed clinical quality along with the manufacturing quality. So with respect to this, recently we have come up with one article of biopharmaceutical risk assessment, which provides a general framework for CBA evaluation for the generic regulatory submissions. So this is a flow chart which we have proposed in that publication. And based on the initial formulation risk assessment, the idea is to identify initial critical viability attributes. So when we are defining the initial critical viability attributes, we can consider a couple of factors as defined here. So in terms of BCS class, as we all are aware that BCS class 1 represents low risk, and class 2A, class 2C, and class 3 represents medium risk, and weekly basic drugs of BCS class 2 and class 4 represents a high risk. Coupled with this, when we talk about the formulation specific to the generic formulation development, when we have IR conventional formulation with the equivalent composition as that of innovator, it can represent low risk. But in case of enabled formulation with non-infringing formulations, then they represent high risk. In case of modified release formulation, due to the complexity, the initial risk itself can start with medium. But then if we are designing a generic formulation with entirely different composition or the formulation principle as that of the innovator product, then it goes to high risk. In terms of the historical knowledge, typically the innovator companies publish a lot of data as a part of biopharmaceutical risk assessment, as a part of public assessment reports. So we can utilize all that data coming from assessment reports or from the patents and literature to understand if any of such attributes are studied and then what is the impact. Along with that, apart from the pivotal data, we also have the data coming from pilot studies. The pilot studies also gives a lot of information in terms of pharmacokinetics of the molecule, intrasubject CV, the availability of biorelevant media, and the ranges of all of these attributes studied. So together with all of these parameters, we can do some initial risk assessment, and then we can define the initial CBS. Followed by that, once the initial CBS are identified, the final CBA risk assessment can take place through in vitro studies, wherein we can use the PBBM modeling, or we can also give with in vivo studies, directly conducting in vivo studies with the ranges of these parameters to understand the final impact. So once we have final CBA risk assessment has been done, then based on this analysis, we are going to define what are the final CBA attributes. So to demonstrate this particular aspect further, we have come up with a detailed case study. So in this case, the drug product is an extended release formulation, and initial risk assessment as per the flow chart that we have discussed has identified that polymer viscosity, polymer grade, and APA PSD can be material attributes which can impact in vivo behavior. And under the process parameters, percentage fluid uptake, and then compression force, and under the formulation variables, the polymer levels and the ratio between polymers are considered as initial attributes. And considering this initial risk assessment using the QC media, which is also a biopredictive media in this case, detailed in vitro evaluation has been performed as indicated in this figure. So we can see in the middle of this figure that we have pivotal reference and test formulation, and this particular extreme profile represents the low level of polymer 1, and this extreme level of blue color line represents high level of polymer 1. And in between, we have the profiles coming from different compression forces or different fluid uptakes. So once we have this biopredictive dissolution data available, then we identified that none of these parameters are impacting the resolution. But in case of critical process parameters, both fluid uptake and compression force have impacted the resolution. And similarly, in case of formulation variables, the polymer level 1 and polymer 1:2 ratio have impacted the resolution. So to further assess the impact of these variables as to define whether they are critical viability attributes or not, we have evaluated the PBBM modeling approach in this case, using the dissolution data that is presented in this slide. So these are the results from the PBBM modeling. And we have compared and assessed the bioequivalence in terms of pivotal test formulation and against pivotal reference formulation. The model validation has confirmed that the model is able to predict the outcome of the pivotal study. And what is interesting to see in both the cases is that the model predicted the edge of failure or bioinequivalence with respect to different attributes. Say, for example, in case of low-level polymer, the model predicted the BE failure on the higher side, whereas in case of high level of polymer 1, polymer 1:2, and then high compression force, the model predicted the bioinequivalence, which is failing on the lower side. So using this particular data, we have done the final risk assessment to conclude the critical viability attributes. And in this case, we have defined the final assessment through 3 different parameters. And the nomenclature, as you can see, probability, severity, and detectability are similar as that of the nomenclature that is used in the QBD. but then we have come with a scoring of low, medium, high without giving a specific numbers to these attributes. So in terms of the probability, we can define this as has it ever happened in the past, but considering the generic development, we typically manufacture the formulation with some history, but then in some cases there might not be availability of history, so we kept this as low. And in terms of severity, it can be defined as what is the impact on in vitro and in vivo performance. In cases like polymer level 1, in case of polymer 1:2 or compression force, the severity is found to be high because they have led to bio-inequivalence. And the impact is both on in vitro and in vivo impact is studied through PBBM modeling. In terms of detectability, in this particular case, the detectability is high because all the manufacturing changes are reflective in the resolution. And the QC media in this case is also acting as a biopredictive media. And then that's where the detectability is high. In terms of the final severe risk assessment, although some attributes have impacted the bioequivalence, but based on the levels that we have studied in this case, we have identified the suitable ranges of all these parameters, which will not lead to bio-inequivalence. And same data also has been a part of our manufacturing batch record. That's where there is a reduction in the final severe risk assessment. So this is a summary of the article that we have published, which is talking about generic workflow for evaluation of CVS. Then the second case study talks about the impact of faster dissolution profiles on the safety. So all of you are aware that traditionally we use the PBBM modeling from the efficacy perspective. In the sense, we use the PBBM modeling to understand the bioequivalence failure or justifying the dissolution specifications. So in this case, we got an interesting query from one of the regulatory agency. The product is an extended release formulation containing an anti-epileptic drug, and the product exhibited faster dissolution profiles during stability, especially at the accelerated condition. And then agency asked us to evaluate the impact of faster dissolution profiles on the safety. So safety becomes pertinent in this case because the drug product contains an anti-epileptic anti-drug. So any changes in the dissolution can have an impact on the safety. And that's where agency asked us to evaluate the impact of these faster dissolutions. So to support this particular query, we have utilized the PBBM modeling. So on this particular slide, we can see the pivotal test and reference formulation. And the brown color line as well as the pink color line, they represent the extreme dissolution conditions or the dissolution data that have been observed during the stability. And along with this extreme faster profiles, we also have dissolution profiles at the boundaries of the dissolution specification as per the USP chapter. So these specifications are designed as per USP 711. And the impact of these faster dissolutions as well as slower dissolutions, we have evaluated through PBBM modeling. So again, we followed the general methodology of the PBBM modeling. So first, we developed the model and then validated against the pivotal study data. And the model has clearly attributed in terms of the variability that is observed from the study in both fasting and fed condition. And then using the developed virtual population, we have given the dissolution data coming from extreme boundaries of dissolution specification as well as from the stability data. And then we predicted the highest Cmax that is observed among all the virtual populations. So what we can see from this particular slide is that in terms of upper stability profile, or even with L2 and L3, the specifications at upper dissolution profile, we can see that the concentrations have increased as compared to the pivotal test and pivotal reference formulation. But in all the cases, the concentrations are significantly lower than the reported safety levels. So the reported safety level for this product is 100 microgram per ml. And as we can see, in all the cases, even with the extreme dissolution profiles, we have observed the highest Cmax, which is significantly lesser than maximum safe concentrations. So eventually, we submitted this justification to agency, and then we have clarified that there is no impact of faster dissolutions on the safety of the drug product. The third case study, I think many of the generic companies sitting in this presentation can correlate, is to evaluate the gender impact through PBPK modeling. So again, recently, a lot of generic companies are getting these particular queries related to understanding the impact of different genders on the bioequivalence of this product. So typically, especially in India, due to the socio-cultural reasons, it may not be possible to include the equal number of female subjects in the bioequivalence studies. And that's where majorly male subjects are included in the bioequivalence calculation. But then as a drug product is intended for, most of the drug products are intended for both genders. These days we are getting some of the common deficiencies to understand or to extrapolate the bioequivalence results obtained in one gender to the another gender. So in this case, we got this query for IR drug product containing a BCS class 2 molecule. And then as I described, agency has come up with this query. So we utilize the PBPK modeling in this case to address this query. So we developed the PBPK modeling with detailed input of all enzyme transporter kinetics. And then we have done the thorough validation against the pilot as well as the pivotal data. And then subsequently, we utilize the model to predict the exposures in of males and females. And as we can see that the reported data in terms of ratio of the males to females has nicely matched with our predictions. And thereby we submitted this model along with the literature data to the agency to support the absence of impact of sex on the bioequivalence study results, and the fact that bioequivalence results in one gender can be extrapolated to another gender. This is the case where we have some literature data as well as we supported this through modeling, but this can be of significant utility in those cases where there is no sufficient literature information with respect to gender impact. Then the last case study is with respect to demonstrating the discriminatory power of the dissolution with DDD+. So in this case, the drug product is a BCS class 3 API with a drug load of 60%. And there is no discrimination that has been observed in the QC media. And hence, agency asked us to justify the discriminatory power of the dissolution method. So 2 points to be noted here. The drug product contains BCS class 3 API, that too with a drug load of 60%. So which means the changes in the polymer may not be impacting the dissolution rate, and because of the high solubility of the drug coupled with the high drug load. So we tried a few experiments using the same QC media, but then we are unable to understand the discrimination. So we thought, can we go to extreme boundaries of the excipient that are used in this extended release formulation? And then are we going to see some discrimination? But then when we went to the extreme levels of excipients, we are unable to manufacture the drug product itself because of the low polymer concentration. So that's where we have utilized the DDD+. So first we developed the DDD+ model for the reference as well as the test product. And then we designed the virtual batches with different levels of polymers, starting from 90% until 30%. So what we can clearly see is that when the polymer level is 50% of the current formulation, it is going to lead a failure of F2 when it is compared with the pivotal test. But unfortunately, these formulations we are unable to manufacture because of the low level of polymer and the manufacturing constraints. But then we are able to demonstrate through these virtual simulations that the QC media is of discriminatory nature, especially when we go to the extremely low levels of polymer. So this is how we have utilized DDD+ in case where we are unable to manufacture the batches with respect to the practical constraints. So these are some of the case studies, and in the next table, we would like to talk about our experience of specific regulatory queries that are received on the PBBM models, and then how do we try to adjust these models. So some of the queries are related to justification or optimization of the model input parameters, especially effective permeability. And then in those cases, to address the queries, as I discussed before, either you need to have the experimental or literature data, but in absence of that data, we can do parametric sensitivity analysis to demonstrate the suitability of the optimized value. And then there are few questions on the utility of Z factor. It has some inherent issues, as all of us understand. So then typically we try to answer it through demonstration of the Z factor in relation to the absorption dissolution curve. And then we try to demonstrate the calculation method and the impact of solubility, time points, the fit, and etc. Then the question is related to dissolution method and the biorelevancy and the ability to reject non-bioequivalent batches. So this aspect we have discussed in detail in the previous slide. It is ideal that the QC media represents bioprediction, but in absence of that, we need to have additional data coming from a separate method to answer this particular query. And most important query that we are getting in recent times is the ability to provide the mechanistic framework of the model, which means is the model is able to demonstrate the entire ADME processes. So in those cases, what we observed is that PBPK, when we combine with dissolution data input, that can provide the entire mechanistic framework. But then we can also utilize the mass balance diagrams, especially what is listed in the EMA PBPK guidance, to talk about the mechanistic framework of the model. Then recently we are getting a lot of queries with respect to critical viability attributes, which we have discussed in the past slides with the help of case study 1. And then validation against the failed BE data is also another query to demonstrate the robustness of the model. But in some cases, we may not have the failed bioequivalence data that is available with us as a part of the product development. So in those cases, at least we can demonstrate the sensitivity of the model towards the identified formulation attributes or the dissolution data. Then in some cases, we all agree that the PBBM modeling itself is unable to justify the regulatory query. But then in those cases, we can use the totality of evidence approach, which includes the biopharmaceutical risk assessment and the multimedia dissolution to answer the regulatory queries. Then again, different release dates and corresponding IV/IVC. So ideally, it is important to have, especially for the MR formulations, different polymers and the release date. But as the generic development focuses on obtaining the bioequivalence formulation, this kind of data is not available. So if agency is asking with respect to this particular query, it might be a little challenging. But then again, we can demonstrate the sensitivity of the model towards the dissolution. And we also have seen with the fourth case study, how do we utilize DDD plus to demonstrate the discriminatory power of the dissolution. And with the third case study, how do we utilize the PBPK modeling to understand the impact of gender on the bioequivalence study results. So these are some of the regulatory queries and how do we address them. So with this, I would like to end my presentation with a couple of concluding remarks and future thoughts. So throughout this presentation, we have seen that PBBM modeling has wider avenues in terms of generic product development. And as a generic company, we also have seen that other regulatory agencies like ANVISA, New Zealand and China, they're also open to modeling-based justifications. We have, I think, thoroughly understood the application of PBBM modeling with respect to F2 mismatch, dissolution profile dissimilarity, and the biowaivers, etc. But in future, we can see that more and more focus is being made in terms of biopredictability of the QC media and the mechanistic framework of the PBBM models. And I think in future, the focus areas will be in terms of waiver of the FED studies and multiple steady state studies also PBBM can be utilized. Most importantly, the takeaway from presentation can be the CBA evaluation framework, which we have presented, which can facilitate the generic regulatory submissions with respect to CBS evaluation. So overall, I think I'm happy to say that PBBM modeling is increasingly being recognized, and most of the regulatory agencies are open to these kind of submissions. And through workshop like this, this is the best way to share our knowledge in terms of regulatory queries or the model development and model validation, so that we leverage the power of PBBM modeling to the fullest. So with this, I would like to thank Dr. Tausif Ahmed, who is heading the biopharmaceutical and bioequivalence team at Dr. Reddy's, and the global clinical management for the support, and Dr. Reddy's management for providing all the infrastructure and the support, and all the cross-functional teams and the collaborators and the regulators who have contributed to the data generation and the feedback. And last but not the least, I would like to thank organizing committee, especially Tycho, for giving me this opportunity and for inviting me to speak in this particular workshop. So these are a couple of publications from Dr. Reddy's, which we have published in the recent 3 years in the area of PBPK and PBBM modeling. And these publications list is a part of the presentation. So if you are interested, please go through this list. So with this, I would like to thank everyone for your kind attention. I would like to thank organizers for giving the opportunity, and looking forward to hear from you. Thank you.
Thank you, Dr. Kolipara. I think we'll allow a couple of questions, maybe 2 questions. All right, there's one question for you.
Hi, I'm Russ Rackley with Beatrice. I was just curious, how do you or can you incorporate reference lot-to-lot variability in defining a safe space? Is that ever considered?
Yes. So typically when we develop the model, we validate the model with the data that is coming from the pilot formulations, and there might be cases where we have used a different reference lot in the pilot studies as compared to the pivotal studies. I think through the dissolution data of the reference lots that are used in the pilot and pivotal studies, the variability is already considered in the model. And further, whenever we do the virtual bioequivalence simulations, one of the variability contributing factor is coming from the dissolution parameters. In case of IR formulations, it can be Z factor. In case of MR formulations, it can be Weibull parameters. And that's where the incorporation of variability of these dissolution parameters will also take care of the variability of the dissolution profiles. So in summary, to answer your question, the variability of the reference formulation lots is already embedded as a part of dissolution, and further it's incorporated during the virtual bioequivalence simulations.
I mean, you run into cases occasionally where you have to resort to using multiple lots or different lots from time to time. And occasionally you run into a case where, while marketed lots are considered currently therapeutically equivalent out there, they may on a PK basis not necessarily be conclusive with respect to bioequivalence. So that alone seems like it would define some kind of safe space without doing any modeling.
Yes, but then maybe a couple of examples I can give. As you are aware that the reference lots from different markets, and for that matter, within the markets can have different dissolution profiles. And there can be cases, especially for BCS class 2 molecules, that the bioequivalence is based on the dissolution profiles. So in some cases where we observe the difference between reference lots, then we try to choose a reference lot which has got a closer dissolution profile as that of the test product. And we further evaluate the bioequivalence or bioequivalent risk assessment by incorporating the dissolution data of this different reference lots in the PBBM, and we try to assess the bioequivalence. But I agree with your point that different reference lots can have different dissolution behavior, which can also act as a safe space. But in our experience, the bioequivalence assessment not only depends on the test product, but in some cases, the reference lot selection and the suitability of the dissolution profile is also going to govern the bioequivalence. So that's how I answer it from the generic product development.
Okay, thank you. One other comment is with gender analysis, provided the study is set up as a crossover, I don't think there's any real evidence out there that I'm aware of, strong anyway, that suggests that there's a gender by formulation fraction typically occurs. I don't know if that's something to think about, but these are just some thoughts I had.
I agree with you. I think modeling approach for evaluation of gender impact definitely can act as a supporting evidence. But are we going to rely completely on the modeling-based approach for the gender evaluation? That can be still questionable because that physiology cannot be entirely mimicked by the systems. There are certain limitations at this point of time. But definitely it is going to act as a supporting evidence for the gender impact evaluation.
All right, one more question, Rob.
Sorry.
I'll be quick. I'm intrigued by the fact that you use DDD plus to justify a method that is not otherwise discriminating. I wonder, can this be accepted using this modeling to justify otherwise non-discriminating dissolution method? This may be a question for some of our agency friend here. Is it considered an acceptable approach? Or maybe you exhausted within maybe another alternative. If I exhausted all the possible mean, and I'm going to say, I just couldn't discriminate because this is BCS 1 and 3 compound. Why do I want to kill myself so much just to show for the sake of discriminations?
Yeah, I think in this case, this is a BCS class 3 molecule, but this is formulated as an extended release product. So that's where, despite the high solubility, there is a dissolution profile over an extended period of time that is coming from the polymer. But because of the fact that the formulation has 60% of the drug load, the release is majorly governed by the 60% drug load containing high soluble molecule, but minimal contribution is coming from the polymer, which is occupying almost 20% or so. So that's where we are unable to get the discrimination in the in vitro or the QC media condition. In the practical ranges of the polymer, when we reduced up to 10% or 20%, there is no discrimination. So to support the fact that for this extended release formulation containing BCS class 2 molecule, the method is still discriminative. That's where we have generated these virtual profiles, and which has shown that 50% or 40% of the polymer will definitely lead to F2 mismatch. But as I mentioned, these batches are difficult to manufacture because the polymer quantity is so low. So in a way, we try to summarize that the discriminatory of the ability of the dissolution media that is there, but it is not being evident because of the manufacturing constraints.
So in the interest of time, I think we're going to stop the discussion here. Sorry, folks. I'm sure Shivacharan can answer your questions over email. Feel free to bug him. So thanks again, Shivacharan. Appreciate the great talk.
Thank you
Our
very
next
much.
speaker is Fong Wu from Office of Generic Drugs, FDA, and Fong is going to talk about OGD's perspectives on PBBM. So Fong, please go on.
Good morning, everyone. The topic of my presentation, I'm going to talk about OGD perspective on PBBM applications for generics. Here is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. The outline of my presentation today, I first will talk about regulatory questions that PBPK absorption modeling can help answer. Today, in most of my presentation slides, I use PBPK absorption modeling, and the science is similar, and some case examples show the biopharmaceutics applications. Those are the PBBM. General PBPK modeling procedure in ANDA submissions, then followed by the PBPK guidances supported by regulatory applications and research. Then I will talk about 4 regulatory case examples of using PBPK absorption modeling, PBBM in Office of Generic Drugs, and then talk about the research highlights of the modeling and simulation to support guideline. Regulatory questions that PBPK absorption modeling can help answer include evaluated impact of changes critical quality attributes, setting dissolution safe space, evaluate the risk of formulation mechanisms change, and simulate in vivo alcohol dose dumping, and also include using the PBPK absorption modeling for the impact of food on BE, the impact of gastric pH change on BE, and simulate GI local concentration to support BE assessment for GI locally acting products, and the wave of in vivo studies, and then also evaluate the BE in specific population, including pediatrics. The then general PBPK modeling procedure, as we listened these 2 days and the previous presentation, we all know this procedure. The model input can include the in vitro data of test and reference formulation and the clinical data sets. And first, we use IV data, ideally use IV data to develop a disposition model, then use oral data to develop absorption model. After verification, validation of the model, the sensitivity analysis can be conducted to identify the key factors that impact in vivo performance. And then the virtual BE simulations can be conducted to set clinically relevant critical quality attribute specifications and simulate BE trials between reference and test product. So today I'm going to give you some highlights of the utility of PBPK absorption modeling that impact regulatory decision-making in OGD. The case example include using the modeling to do the risk assessment of the impact of particle size distribution on BE and set clinical relevant 3-tier PISD specification. And the risk assessment for the deviation of dissolution profiles on BE, then conduct a risk assessment of non-comparable dissolution on BE for ER product, and then identify biopredictive dissolution and support BE evaluation by using the PBPK absorption modeling. The first case example is using PBPK absorption modeling in assessing the impact of a particle size distribution on BE. This is a capsule product. The PK parameters, including the Cmax and AUC, are found to be sensitive to changes in mean particle size of drug A and the fasting condition. The applicant submitted a magnetic absorption model to link this particle size distribution with in vivo data. Here is the workflow. The model input include the experiment measured physical chemical parameters like log P and pKa, and they use the in silico parameters, solubility biosalt, solubilization ratio, and of course the particle size is measured and input as an input in the model. And they used a population PK informed clearance into the model and build the initial model. Then they have a rich data set of different batches with different particle size and from the 3 different strengths and the fasted and the fat conditions for model development. And then they used the data set not used for model development for model validation, and then get the model for further application. After the review, and we use the model and to simulate, and with fixed D50 and changed D10 and D90 for different range. And based on the model simulation, we found that there is a low risk of having a non-BE by fixing the D50, but changing the D90 over a relatively wide range. So these PBPK modeling simulations suggested that test versus reference PK matrix showed a low risk of non-BE when D90 changed over a wide range with a certain fixed value of D50 for all strengths. The modeling results support a satisfactory BE assessment of this ANDA and support a tentative approval of this ANDA and setting a clinically relevant 3-tier PSD specification. The second case example is using PBPK absorption modeling to support a waiver for lower strength of oral tablet product. This is an IR product. Some background information. We know waiver of lower strength can be dependent on formulation, proportionality, dissolution similarity, bioequivalence, and other strengths. However, for this case, the lower strength of this drug B has a faster dissolution profile compared to biostrength, which is a higher strength. Per RD label, the drug product should be taken with food, and there is a positive food effect for this product. The applicant used a magnetic absorption model to predict whether the faster dissolution of lower strength has significant impact on the in vivo performance under fasting condition, but not under fat condition. So this is the general flow chart for the model development, validation, and application. The applicant developed disposition model and estimate disposition parameter using PK profile of RD at higher strength. Then they incorporate the in vitro dissolution data and also use PK of RD at higher strengths to develop absorption model and estimate absorption parameters. And for validation, the applicant used the in vitro data, dissolution data, and PK of both test and RD at higher strengths, and to predict the PK, and also conducted the virtual BE between test and the reference at higher strength. The validation showed that the prediction error less than 10%, and the population simulation can capture observed data variability, and the test product RD at higher strength is predicted to be BE, which is captured observed result as well. And then after the validation, the applicant used the dissolution profile at a lower strength as input for test. They assume this faster dissolution profile also occurs for higher strengths. Then they incorporate the dissolution of test pivotal bio batch at higher strength as reference. Then they conduct VBE simulation under fasting state. Then simulation predict BE. So we reviewed this submission, and deficiency identified are like we recommend, and we also provide a recommendation. We recommend that the applicant validate the model for the intended purpose using different strength or using data from formulation with different release rate. As you can see, they use just the R and T, they have the similar release rate. And they need to demonstrate prediction performance for PK data of biostrength and the fat condition, because this product is taken with milk. In summary, PBPK PBM modeling and simulation was used to evaluate the impact of faster dissolution profile of lower strength compared to higher strength on in vivo performance. The model should be sufficiently validated before being used to evaluate such impact. The third case example is using PBPK absorption modeling and IVIVC to assess the impact of non-comparable dissolution profile of an ER product on in vivo performance. Some background information. Both ROD and test product use osmotic pressure to deliver drug C at a controlled rate. And then per ROD label, the drug product should be taken with food. For this case, the dissolution profile of the lower and higher strengths of the test product are comparable to that of the bio batch, which is middle strength of the test product in both QC and multi-pH media conditions, but not comparable to those of corresponding strength of ROD. The applicant developed both mechanistic absorption model and IVIVC to justify that TR ratio for Cmax and AUC of all strengths remain within the 80 to 125 limit. This is the workflow for the model development, validation, and application. The applicant developed disposition model and estimate the disposition parameter after using the PK profiles after IV infusion and IR tablet. Then they developed the absorption model and estimate absorption parameters using in vitro dissolution data of ER capsule and tablet in QC medium. And then they use the in vitro dissolution data of ER capsule and tablet with multiple strength in QC medium to validate the PBPK model. They also use the literature data to do the validation. But then they didn't use this PBPK model for further application. They further developed the IVIVC model. Dissolution profile of multi-medium at middle biostrengths as input for two test formulations and ROD. They use these dissolution profiles for develop 4 IVIVC model, each for one specific pH condition. After that, they validate the IVIVC model by using the dissolution profile in multimedia at middle biostrength as input for 2 test formulation and ROD, and using also, of course, using relevant PK profiles to validate this IVIVC model. Then they use the IVIVC model to predict fraction in vivo release and predict test versus reference result for AUC and Cmax by incorporating the dissolution profile in multimedia at lower and higher strength as inputs for test formulation and also ROD, because the aim is to support the waiving for the study for the waiving the lower and higher strength. After review, we found that the for IVIVC model, because the middle biostrength has similar dissolution for test and R in QC medium and the multi-pH medium, the applicant's IVIVC model were not developed based on formulation with different release rate. even based on the applicant's IVC model, despite the above deficiency, the predicted PR ratio for Cmax for lower and high strength in multi-pH media still falls outside the 0.8 to 1.25 BE limit, which indicates the applicant's IVIC model failed to support that generic product is BE2 already. We also provide the recommendation for PBPK. There is a lack of non-BE batch to challenge the PBPK model. The applicant is recommended to use available and conduct a sensitivity analysis to demonstrate the biodiscriminating capability of the model. The developed PBPK absorption model with further sufficient validation, together with virtual BE simulations, may be used to provide risk assessment and support the justification for the non-comparative dissolution of both higher and lower strengths of test and RLD products in multi-pH media. In summary, we know that for this IVC model, they were not developed based on the formulation with different release rate. This limitation restricts its utility to evaluate the effect of non-comparable dissolution, and develop the mechanistic PBPK absorption modeling with sufficient validation together with virtual BE simulation may be used to provide risk assessment. The fourth case example is using PBPK modeling to support BE evaluation of GI locally acting product. This drug is a delayed release tablet. It is indicated for colitis. The product-specific guidance for this product recommends a fatting PKB study and a fat PKB study and comparative dissolution studies at 4 different pH: 6.5, 6.8, 7.2, 7.5. However, the F2 values for dissolution profile comparison between test and RLD were less than 50 at pH 6.8 and 6.9 buffer condition. Based on the clinical study, the two products were found to be bioequivalent for systemic PK and both fasting and fat condition. The PBPK absorption modeling was developed by FDA reviewer and used also by FDA reviewer to predict the local drug amount in the colon by incorporating dissolution data at different pH conditions for supporting the decision making. And here is the workflow. The IV PK data obtained from the literature were used to estimate disposition parameter. And then the PBPK model was developed for this drug DDR tablet by incorporating 3-stage dissolution data using pH 6.8 or pH 6.9 or pH 7.0, pH 7.2 as stage 2, as stage 3, and incorporated into PBPK. The stage 1 is hydrochloride medium, and stage 2 is the pH 6.5. That is the available data. And then the PBPK model was validated using the dissolution and PK data obtained from clinical BE studies of this product and other products. And we then use the dissolution data at different pH conditions, first to evaluate whether the dissolution data at certain pH is biopredictive. Then use this model to predict, and also the biopredictive dissolution to predict local drug amount in the colon, and also conduct a population simulation to compare the predicted percentage of drug absorbed in the colon for T over R and support BE assessment. The simulation results showed that dissolution profiles at both pH 7.0 and pH 7.2 at stage 3 is biorelevant or biopredictive to the PK profile. Population simulations show that the percentage of drug absorbed in the column is similar between R and T, with the 90% confidence interval of TR ratio falling within 80 to 125. This table shows the predicted PK parameters for the amount of drug in colon. In summary, this PBPK model suggests that the three-stage dissolution profiles using both dissolution at pH 7.0 or pH 7.2 as stage 3 may be biopredictive to the local and systemic exposure. The PBPK model can be used to predict that the percentage of drug absorbed in the colon is similar between R, ILD, and the test product. So next, a couple of slides, I will introduce our research used to support the other guidance. We know that there are some guidances are published, the PBPK in biopharmaceutical applications and PBPK used for PHDDI. And then these new issued guideline, the ICH M13A in January 2023, talking about some topics I listed here, supported by PBPK research, modeling research. This is a topic included in ICH guideline for fasting and fat study condition. The design of B study with regard to the use of fasting and fat condition depends on the dosing instructions of the comparator product, as well as the properties of the drug substance and the product formulation. The rationale can be supported by modeling, including PBPK modeling and other absorption modeling. This is a research we conducted. We developed a acyclovir model, and you can look at the figure. The model can capture well the PK of acyclovir under fasted and fat condition. Then we also predict using the VBE to predict the BE. It is BE under fat condition and the fasted condition. These research actually were presented in different conferences or the other meetings. This was presented in 2022 APS conference. So I will not talk too much detail for this. This is the first a couple of the research projects, but we know that this acyclovir is a low-risk product. We are right now conducting the research for some moderate to high risk product for supporting the evaluating of the food impact on BE. Another topic included in ICH M13 guideline is pH dependency. For the product with formulation dependent PHDDI, the modeling using the validated PBPK modeling can be used to support assessment of the risk of bioinequivalence. We have a product-specific guidance for palbociclib tablet, actually recommended. In addition to the fasting and the fat BE study, we also recommend fasting study, a BE study in presence of acid-reducing agent to see whether there's still BE under this situation. We conducted some research to support these topic. And this was research. We have a publication, a couple years, 2 years ago, 3 years ago. And we developed a PBPK model for 4 model drugs: tapentadol, darunavir, erlotinib, and saxagliptin. Tapentadol is BCS class 1, and darunavir and erlotinib, BCS class 2 drugs, and saxagliptin, BCS class 3 drugs. And we use the PK data following oral administration without acid-reducing agents, and develop the model and then verify with data from additional PK studies in the presence and absence of food. This developed model, validated model, was subsequently used to predict the extent of drug-drug interactions with ARA co-administration. The model simulation suggests that PBPK modeling developed could adequately describe the lack of effect of ARA on the PK of tapentadol, darunavir, and saxagliptin, and could qualitatively predict the effect of ARA in reducing the absorption of erlotinib. I will not go to the detail. I list the reference in the slide. That is from a new drug side for predicting the pH-dependent DDI. Here I list some considerations for evaluating the impact of gastric pH on bioequivalence for generic drugs. For generic drugs, additional BE studies may be needed. These additional BE studies means that conduct a BE study in subjects with altered gastric pH. This may be needed when there are formulation-dependent gastric pH-mediated DDI. The risk is high under certain situations. For example, when test product and RLD product contain different level of pH modifying excipients. The PBPK model to predict the PPI-based DDI, this is an important step towards identifying formulation-dependent DDI. For these product with formulation-dependent pH DDI, scientific justifications, for example, pH solubility profile, comparative dissolution testing at multiple TPH, and the modeling may be used to demonstrate that BE study in the gastric pH altered situation may not be needed. Here I list a couple of the publications based on the internal and external research. Thanks for our collaborators and also our reviewers and researchers. And then next slide as well lists the other recent publications. You can refer to details when the slides are available later. In conclusion, currently modeling simulation tool, for example, PBPK absorption modeling, PBBM has been increasingly used in generic drug applications. The case examples of PBPK absorption modeling include evaluate the impact of particle size distribution on BE and support setting clinical relevant PSD specification. And the modeling can be used to conduct risk assessment on impact of a non-comparable dissolution profile of test and reference product on in vivo performance. And the modeling, PBPK absorption modeling, can be used to help identify biopredictive dissolution and support the evaluation of a GI locally acting product. I would acknowledge the people listed here and for their hard work and support. And also, I would like to thank the applicants and submit these submissions with the modeling and also our colleagues in FDA for their support. Thank you for your attention.
Thank you, Fang. Questions?
Yeah, I'm clear. Thank you. Oh, is there any?
Morning. Thank you for the presentation. Joe Kirschner from Pfizer. I just had a clarifying question. In the first case study where you were looking at where the case example was the particle size distribution. In the example that you showed, it seemed like they only changed the breadth of the API particle size distribution rather than changing the mean values as well. Can you just comment on whether breadth of API particle size is sufficient for spec justification, or would we also need to look at changes in the mean values as well?
The particle size distributions we incorporated into the model by using mean and the variability, and then predict the PK. So yeah, we need to consider the mean as well as the variability or the distribution. Did I answer your question?
Over there.
Yeah.
Oh, yeah. Okay, thank you.
All right. Thank you, Fang.
All right, our next speaker is Dr. Claire McKee, and she's from Janssen Pharmaceuticals, and her presentation title is Application of Virtual BE Trials to Support Formulation Bridges. Welcome.
All righty. I realize I'm now the person standing between you and coffee and cookies. So I will try get through. No, I will take my time, but at least address, I hope, some of the critical questions that we're looking at, certainly with the industry, with respect to virtual BE trials. So firstly, thank you to the organising committee for giving me the opportunity to give this short talk. So without further ado, let's jump indeed into the application of virtual BE trials to support formulation bridging. So over the next few minutes, I'd just like to have a quick recap on BE versus virtual BE. Then consider some of the questions that could be covered under formulation bridging. And then using two examples, illustrate how we've been using virtual BE, certainly within Janssen for internal decision-making, and then close up with some points to consider. So as a quick recap, certainly within CMC, we use bioequivalence to assess the comparison of the relative bioavailability between two drug products containing the same API. This is usually assessed through a clinical study where we look at the PK comparison. Obviously, there are clear guidelines and clear acceptance criteria, those you are all well aware of. And the aim is to demonstrate that your second drug product is bioequivalent to your reference or your first drug product. Virtual bioequivalence is when bioequivalence can be demonstrated using modeling and simulation, ideally in place of a clinical study. A PBBM is built, and you've heard many examples over the last 2 days, so I'm not going to dwell too long on that, but using all available drug substance, drug product data, and then obviously using the available clinical data to validate the model. Obviously, the PBBM model should be built for the intended purpose. Obviously, it can save overall time and money. It can also hopefully in the future reduce the need to dose additional drug products both to patients and to volunteers. But as we've heard also a lot in the last couple of days, it should be built with the intended purpose. So from a CMC perspective, I'd now like to just move on to sort of areas of potential application. So within CMC, I think the major areas that we do look for BE studies, three main areas. First one is change in manufacturing site or a site switch. Second one is creating or introducing a new formulation or drug product, certainly from an innovator company. And also, as you saw from the first presentation this morning, introduction of a new drug product from a generic company. So diving a little bit deeper into that second bullet point of creating and introducing a new formulation or drug product, the types of sort of clinical studies that certainly CMC colleagues are interested in pursuing or evaluating are a bridge between two formulations. This could either be API, material, platform, or process-driven, or a combination thereof. The second one is evaluating a food effect, obviously based on how your first drug product would have performed in this situation. Third one is evaluating the co-administration of a PPI on your in vivo drug product performance. In effect, this is the DDI of the CMC world, but it's very useful to elucidate population patient-wise. And then last but not least, setting clinically relevant specifications. As you've also heard over the last couple of days, a lot of focus is often on dissolution specifications, but that can also extend out to, say, API PSD, for example. So then diving a little bit deeper into formulation bridging, I'd just like to pause for a few minutes and consider the types of questions that we might be looking at or considering under this category. The first question is which APIs or drug product could qualify for formulation bridging? And actually, it's important to consider when in the project trajectory that bridging would be required. And what do I mean here? Is it pre, post, or during pivotal studies? Meaning, is it a scientific or sponsor risk and question, or is it a regulatory risk or question? The second question that comes to mind is the BCS classification of the drug. Is it a BCS1? Is it a BCS3 API? Here I'm predominantly talking about IR formulations, but is that level of change covered through, say, ICH M9? For BCS2 and 4, then obviously the timing of that bridge is really important. And actually, it's probably where modeling and simulation and even virtual bioequivalence can actually help you look at more drug product understanding or even de-risking. And certainly the types of studies that we've had to evaluate, or certain types of studies we've had to consider within BCS2 and 4, is a liquid to solid bridge. You've got a solid to solid bridge, which could be, for example, a capsule to a tablet change. And there, highly likely, you've got change in excipients and a change in process. The third one is actually a change in quite an amount of excipients. And certainly for BCS3, this could be, say, outside ICHM9. Certainly for BCS2 and 4, outside certain level of changes in SUPAC. Appreciating at this time, your product is still in development, but these particular guidelines can at least help steer your sort of path forward. The fourth one is actually a change of platform. And this is where potentially you might have to move from a conventional to an enabling formulation. Here, enabling, I mean, say, an ASD, where your clinical dose is increased and you actually have to move platform to be able to increase that extent of absorption. And then again, last but not least, exploration of drug product CQAs as part of either clinically relevant control setting or patient-centric control setting. So hopefully, having set the scene, I would now like to share with you a couple of examples from our internal portfolio where we've been using virtual BE to help us in decision-making. So the first example is where we've used mechanistic absorption modeling to assess the impact of drug product changes on the exposure of J&J-X. J&J-X is actually a BCS2 API. The first drug product developed was actually an API in capsule. This was used for both the first in human studies and the phase 2 clinical proof of concept study. Actually, the proof of concept study was really positive. And then, of course, the team turned towards phase 3. And as ever, I hope many of my CMC colleagues in here will smile, as ever, with accelerated timelines. So the request to CMC, of course, was to develop that final commercial tablet. So the next round of formulation development included developing the first prototype tablet, also a blending capsule. And they were actually compared in a relative BA study using the API in the capsule as the reference. That study also included looking at some changes in API attributes. So relative BA study looked quite positive. Tablet came out looking really quite interesting. Then the next round of tablet development or evolution included changes at the API synthesis level. We also had to increase the lubricant, and we also had to change the non-functional coat. So we then used modeling to assess those changes. Were we still on the good path? So I think this sort of flow you've seen in many examples over the last 2 days. So again, I'm not going to dwell on it in great detail, but the key steps we took were obviously the oral absorption model development at the level of the drug substance data and also the drug product. We looked at the PK model development side. We then looked at the parameter sensitivity analysis. We did a model validation, including independent clinical data sets. And then obviously the next step was model application, which was comparing the different formulations using virtual bioequivalence. So this next slide, I appreciate it's a little bit busy, but let me walk you through it. So the next slide shows you some of the model outputs. So the left-hand panel highlights examples from the population simulations versus the observed PK profiles for the blending capsule. So BIC's blending capsule in the top graph and the first prototype drug product in the bottom graph. The black lines are the geometric means of the simulations, and the dashed lines are indicating the 100% probability ranges of the simulations. So we felt that the mean profiles and the 100% probability contours indicate that qualitatively the shape and the variability in the concentration time profiles are captured pretty nicely. So based on that, we went then on to the second step, which is where we compared the changes that I just highlighted in the previous slide of drug product 2, if you will, versus drug product 1. We ran 10 trials, virtual bioequivalence trials. These are actually highlighted in the middle panel. I'm not sure, I hope it's not too much of an eye test to read it. But in conclusion, the 10 virtual bioequivalence trials indicate that we're actually falling within the defined BE criteria of 0.8 to 1.25 for both Cmax and AUC at the chosen clinical dose. So we felt that based on this data, a switch from drug product 1 to drug product 2 is predicted to have little or no impact on absorption and exposure, and therefore it would be of little risk to make that swap between drug product 2 and drug product 1. Okay, moving on to the next example. This is where we use mechanistic absorption modeling to assess the impact of changes in API polymorph on the exposure of J&JY. J&JY is a BCS4 compound, obviously characterized by low solubility, low permeability. We actually had immediate release tablets commercially available when we discovered that we had a new API polymorph which had lower solubility. I can see many of you smiling. So in short, what actually happened was that this new crystalline form had appeared in the drug substance crude crystallization step, that the new form was actually looked like the thermodynamically favored form in the process. So actually we're in a position where could we exclusively say that we could generate that first form, which is actually a hemihydrate. So as I mentioned, because the compound is a BCS4, characterized by low solubility, low permeability, we carried out a biopharmaceutical risk assessment to evaluate whether there could be any differences between the forms that may influence the in vivo performance. And virtual bioequivalence was one of the tools, if you will, in the toolkit that we used. So these were the steps taken in the model building. We did a pretty detailed physicochemical characterization of both API forms in vitro, and that included solubility as a function of pH, biorelevant solubility, function of bile, and also intrinsic dissolution rate. We then built the model, obviously using the registered crystal form, because that's the one we had all the clinical data on. So we obviously used the phys-chem properties, the sort of in vitro biopharmaceutical experiments, biorelevant dissolution. So here, I would like to point out from the beginning that the dissolution input to this model was Z-factor based. And then we verified the model using the PK data that we had. Luckily, we had quite a big data set. We were looking, and we had fasted and fed across different doses, and also a range of API PSDs that had been tested in the, as part of the clinical program. Once we were happy with the model, we then went on to doing an in silico assessment of the new crystal form using the model, and then obviously the biopharmaceutical properties of the new form. So this next slide shows you or highlights the outcome of the model verification and the virtual bioequivalence. Again, a little bit of a busy slide, so let me walk you through it. So on the left-hand panel, you can see two sort of distinct graphs, if you will. The top one is Cmax, the bottom one is AUC. And as a function of the x-axis, you can see the different clinical trials or the different clinical data sets that we had. The blue dots are the observed clinical data, and the red dots are the simulations that the model was producing. So based on this, we felt quite confident that the model was able to predict the outcome of the clinical trials across, like I said, a range of doses, fed and fasted, and also as a function of API particle size, which was obviously within the approved specification range. So once we were happy with this model, we moved on to the second step of then evaluating, if you will, for the new form versus the previous form. So we did the virtual bioequivalence. And if you look at the top table, that's for Cmax and Cmax and AUC for fasted. And then we also did the same evaluation in the fed state. And I hope you can read from the data that the simulations demonstrated or indicated that we were unlikely to have any changes in the oral bioavailability when comparing both forms in the fed and the fasted condition. The graph at the bottom of the middle panel is actually some indication of the dissolution data that we used as input into the model. And those were for the available API batches that we had. And what we thought was that we certainly, to have any detrimental effect on the bioavailability, we would actually have to considerably slow down, if you will, the dissolution rate. So what we actually did was use the model to back calculate a minimum dissolution rate that we would have to see to then be able to observe any impact on the PK. And that, in effect, is the solid blue line that you see in that bottom graph. So based on all of this, we felt quite comfortable that we would be okay if we did, by chance, get some of the second form in the drug product. But we did actually do a confirmatory clinical study where we prepared drug products containing 10, 50, and 90% of the new form. All formulations were bioequivalent, which in effect confirmed the in silico assessment. So having, I realize, walked through the two examples fairly quickly, but conscious, as Amitabha said at the beginning, that we had to keep you all caffeinated to be able to get through the day. I'd just like to close with some points to consider. But actually, when I was reading through my slides again last night, I thought, hang on a minute, most of these points we actually debated to great length in breakout session H yesterday. So perhaps Amin and Vera, we have to look back at the notes that we took yesterday. But anyway, nonetheless, these were the points that came to my mind while we were preparing the slides. So the first one is the number of data sets available to verify and validate that initial model. I think this point came up rather nicely yesterday, also in the panel discussion, also with our regulatory colleagues. And I think we all agreed that actually there isn't a number that we can write down. It all depends on the intended purpose of the model and the question that we're building that model to answer. The next point is the appropriate population size for virtual bioequivalence. Should that be 12, 24, 48? Should it be based on previous clinical experience? Or should it actually be based on, if you were to design a BE study for this drug product, what would that study population size look like? And I think that's actually the position we settled on during breakout session H yesterday. The next point is the number of studies to simulate. Should that be 10, 50, 200? The examples I showed you today are using an N of 10. However, there are situations where we have been asked to simulate more. The third point, now this was a super hot topic yesterday, was the inclusion of inter and intra-subject variability. And here I asked Vera and Amin to keep me right. I think we discussed two main routes of input, if you will, and the pros and cons to each. And the first one was, should one use the variability seen inter-subject of the major PK parameters in the studies? Is that one way to go? The second one was about using and understanding more variability at the level of the physiology that probably actually gives more rise to the inter and intra-subject variability. So I'm certainly not going to rehash that discussion, but I think it would be at least useful. We can go back to, as I said, session H notes. Okay, the last point I wanted to raise is, when can a virtual bioequivalence help actually in the program? And I think it actually depends where you are in the timing in that program, and also whether it's an internal decision you want to make, or it is actually a regulatory discussion you want to consider. So certainly if it's an internal decision, now I just pick an example, you're making a new drug product, you want to introduce it into phase 2. Phase 2 is non-pivotal. You can obviously build your model, validate it next to the data sets you have. And if the model looks rather nice at that point, I realize nice is maybe a wrong word, is looking very good, then you can, as a team, decide whether, based on the confidence in that model, you feel comfortable to be able to make that introduction. The second point is actually from a regulatory perspective, and I'd actually like to come back to one of the points we were discussing with the panel yesterday, and that's about the timing of the interaction and the nature of the interaction that companies could consider when initiating a dialogue with regulators. And I'd just like to put out this idea that obviously when we're first building a model using virtual BE, usually we have quite clearly the question in mind for the model. It could be that during development, we've got, say, chapters A, B, and C of that model. We still know that full development phase 3 is still ongoing, and we're still going to have chapters D, E, and F to come out. But maybe this is indeed an opportunity where we can start to dialogue more routinely with our regulatory colleagues, and at least start to build that confidence in, this is the model we're building, this is the question we're building, these are the data sets we've got. Next stage we're going to have the next piece. We don't fully know whether it's going to pull out, but do you at least agree with our strategy, what we've done so far, and what we could be doing as next steps? And, you know, these could be our acceptance criteria. I think we have to be mindful that if we go in with the question of, will this model be accepted? That's the wrong question to ask at that time. But we could at least ask, are we on the right path? And I think certainly for us as innovator companies, we are writing global files. So it would be super cool if we could actually have that dialogue with 1, 2, or even 3 agencies. And I think today, in the last 2 or 3 days, you've seen that focus of trying to get towards that global alignment that hopefully will come in the future. So with that, I'd really like to thank you for your attention.
Thank you, Claire. Questions? All right.
I hope they're easy ones.
Thanks, Claire, for your presentation. Just to elaborate on the last proposal you were making around, reaching out to regulators early on. Should we propose that as part of P22 section of INDs and NDAs, we have a subsection on PBBM, whether we use it or not, whether we waive it or not? So we could have at least a discussion on the need to develop one model or not to support the biopharm risk assessment and the development of the product. And that could take, place as early as INDs, for example.
I mean, this is where I also look towards regulatory colleagues to help me with this answer, but I wouldn't see why not from an industry perspective. Obviously, if we approach a particular agency, their procedure obviously has to be running to be able to have that dialogue. But I don't see why not, really. But I see Flora's got a microphone, so perhaps she would like to
I'm
contribute.
politely waiting for my turn.
Yeah.
They told me I'm the third on the queue.
Yeah. I wasn't sure if you wanted to comment or you had a question.
Yeah. No, that was exactly actually my comment. I was impressed by the maturity, actually, of the reflection that you were showing on your very last slide. And frankly, I'm telling you, for formulation bridging, we as regulator, at least myself as a regulator, have advised to companies to bridge for some time using this, what you call now virtual bioequivalence. The term was not that one exactly, but this has also come from regulators also as an advice. Now you were asking about interaction. This is a good one. If you are ongoing into your drug development program, you're anticipating to come with this variation or change into formulation, I think scientific advice is the place to come, at least for EMA. And the good news, following up on your very last suggestion, is that there is a parallel scientific advice procedure for EMA and FDA. So it is possible when you ask for scientific advice as a sponsor to say, for this advice, I want this parallel procedure. So we will be sitting in the same room with our FDA colleagues, responding exactly the same question. I cannot promise that in the end we'll always be in agreement, but at least we will be informed.
Yeah.
We know exactly on which point we disagree or agree, and we have a forum, very transparent when we discuss. And I have the impression that this is a scheme which is underused for now by sponsors. And each time I'm involved in this kind of procedures, I always find it very enriching in terms of scientific level. So this is something I could also recommend. And the last thing I would recommend is that if you do have several products in your pipeline that would actually use the same approach for bridging, and you want really to discuss at scientific level, at EMA, you need to come. I would advise to come for qualification. Advice. You could say, this is our plan. We have 3 of these products. We're using exactly the same platform to bridge from this formulation and that formulation. How can we, use the knowledge that we're generating in the different programs and have a sort of overall plan this is doable and has been done. So there are paths to interact. The earlier the best. Don't wait to have the data to come. This would be actually my advice. Thank you.
And at FDA, we are also open at IND stage. If your question, are we on the right path? So we may not be able to give you a regulatory decision at that point, but we can certainly have the scientific discussion.
But so, I mean, at the end of the day, if your model isn't completely built, I don't think we can ask the question, will this model be accepted? Because it's not fully there. But no, appreciate the advice. Thank you very much.
Thanks, Claire, for a really nice presentation and also sharing two very relevant examples. I was hoping you'd shed some light on the internal discussions that you had that led to the selection of the FASIV dissolution test as an appropriate biopredictive input to the model. I think it's for your second example, it's a BCS4.
Yeah, no, indeed. No, it's a very good question. And actually what we saw in the in vitro data, I mean, it's a super BCS4, this thing. But interestingly, it's completely, pretty much insoluble in water, irrespective of the pH. However, we do have a pretty big biosolubilization kick with this thing. Obviously, from the first form, which is the hemihydrate, we've done some clinical studies, including sort of regular and high-fat meals. We didn't see the influence of the high-fat meal in the clinic. So we actually chose the FASIV data because it was the most discriminatory system, if you will, or the discriminatory case that we would be in, because actually the second polymorph was about 40% based on in vitro, 40% lower solubility than the first form. So we used the FASIV because it was the most discriminatory setup together with the highest clinical dose to do the evaluation. I forgot to mention that. So thank you for picking it up.
Thanks, Claire. That's really useful.
All right. With this, we'll break. Please feel free to bug Claire in the break, and we'll reconvene at 10:30. So 20 minutes. Thank you.
So our next speaker is Min Yong Woon from the Office of Clinical Pharmacology in FDA, and she's going to talk about applications of PBBM in Clin Pharm. So, over.
Thank you for the introduction, Amitabh. I just want to make sure that everybody can see me because I'm very small, so I'm trying to reach the microphone as low as possible. Okay, so we are going to switch a gear a little bit in this presentation, and we will be talking about the utility of the advanced oral absorption modeling in clinical pharmacology assessment. So here is the disclaimer that I'm talking about my opinion, and you should not consider this as a FDA's opinion, official positions. Okay, so the goal of the presentation is to share the utility of the advanced absorption modeling for clinical pharmacology assessment. So we've discussed a lot about the application of the advanced oral absorption modeling for drug quality and formulation development in the past 2 days. The same tool, the PBPK oral absorption modeling, is a useful, one of the useful tool sets for clinical pharmacology assessment for PBPK analysis as well. So the goal is to share some case examples with a special emphasis on sharing the current application areas and also sharing insight into what's the potential application or expansion of the oral absorption modeling to the clinical pharmacology decision-making areas. And we want to also share that what we view as a current gaps and what's the improvement and the further research that may be desired for in terms of the further developing the oral absorption modeling tools to expand the application areas. So us begin with the overall picture of the PBPK application in the clinical pharmacology assessment. So on the left side, you see the number of NDA submissions containing PBPK proposals. So you can see, indeed, there has been a rapid increase in the PBPK analysis over the last 2 decades or so, and it's becoming a routine part of the NDA application. And the areas of application also rapidly changing, expanding beyond the, don't know what's the, maybe I will not figure out how to use the pointer. So you see on the right side, beyond the traditional application to the DDI evaluation at the systemic level. So for those newer application areas, we view that oral absorption modeling is one of the important tools to apply. So reflecting the expansion of the PBPK analysis in the new drug development, there are multiple guidances. I'm showing here is FDA guidances for industry, mentioning the PBPK. Definitely, this is not the entire picture of the PBPK opportunity represented in the guidances. So we do recommend early communication with the agency. One thing I want to highlight in the most recent guidance, the biopharmaceutics application, is that the drug product quality and mechanistic understanding of their interaction with physiology to affect in vivo drug exposure. This foundation principle applies to both biopharmaceutical application and also the clean farm assessment decision-making application as well. So we are talking about the same tool for different type of decision-making. So, sorry, here in this presentation again, we will be looking at those other opportunities. So when to leverage advanced absorption modeling for decision-making, regulatory decision-making from a perspective of clinical pharmacology assessment. Obviously, when we need to mechanistically characterize complex oral absorption matters, they are arising from the interplay of drug physicochemical properties and formulation properties, as well as physiological factors. When we need to characterize those interactions for making a decision about how significant of the PK findings I have in terms of the intrinsic factors and extrinsic factors affecting the investigational drugs PK. So here I list some of the example areas that we have been using the oral absorption modeling. Some are more mature, and we have seen these areas of application in the NDA submission, and some are more, let me say, young and emerging areas. And I see that there have been a lot of development in the tool development in the oral absorption modeling in this workshop. Okay, so here are the case examples. I will be using 3 groups of case studies to share that, discuss about the opportunities in the clinical area. So first one is pH DDI, drug-drug interaction evaluation. Basically, what's the elevated gastric pH during absorption when co-administrating the drugs with acid-reducing agent. And we will be looking at some of the examples in terms of the providing support to pediatric PK evaluation, and also a very new area of addressing the excipient, potential excipient interactions. So the first case is the duvelisib. about the pH-dependent DDI potential variation. So in this case, the PBPK objective was to evaluate duvelisib exposure change because of the elevated gastric pH due to the concomitant use of the acid-reducing drugs, while accounting for the effect of drug particle size differences. So some of the background is that, so by the way, since the major focus is sharing the case examples, technical details about the modeling and the input parameters may be a bit high level, but I provided the references to the published FDA reviews. So please do consider reading those reviews if you want more information. Okay, so duvelisib solubility is pH dependent, which decreases with increasing pH, indicating the potential pH dependent DDIs. And elevated pH may lower the duvelisib solubility and therefore the exposure in vivo. And for this drug, drug particle size is a critical matter, critical material matter that can affect dissolution and ergo drug exposure in vivo. So the applicant developed and submitted PBPK model for two purposes. One is for biopharmaceutical application in support of the setting the particle size distribution specification. And for that purpose, the model was acceptable. And the other purpose was to address the clinical pharmacology question about the pH DDI potential of this drug product. So this is the PBPK model development, validation, and application workflow. So while we are not going into the detail, but I want to emphasize that the key input parameters, how the model was developed, put together, the structure and developed, included parameters, and how the model was validated was all focused on this PBPK analysis objective, which was to assess the elevated gastric pH, what's the effect of this change on duvelisib exposure, while we are considering the biopharmaceutical properties of drug product, the particle size distribution. So you can see the input parameters, the pH-dependent solubility in aqueous as well as biorelevant media, and also the in vivo PK studies using different particle size distribution. And those were fed into the PBPK model, and we review the model validation from that perspective. So basically, it is integrating the information about the duvelisib solubility and dissolution, several pieces of information, and PBPK model translated those information to show what is the expected in vivo exposure in terms of the PK parameters that we review. So here is the model application briefly about the result. So this PBPK model was used to predict the duvelisib exposure change for market image formulation, as well as a hypothetical formulation that has particle size distribution at the upper limit. So this is the risk assessment added by FDA reviewer. And the model predicted what's the expected exposure change because of the elevated gastric pH, the absorption site condition change on the exposure difference, resulting in the difference in exposure. So overall, the predicted effect was considered not clinically meaningful in this case. And the impact of duvelisib absorption modeling is that the model was acceptable as we briefly look at the purpose and the input of the parameters and the available data for validation was acceptable for this purpose. And also this model was used to support the risk assessment and the overall regulatory providing support to the overall regulatory decision about the concomitant use of this device with the acid reducing agent. Okay, the second case is also about the pH DDI potential evaluation for asciminib. So in this case, PBPK objective was a little different from the first case. In this case, the goal of the PBPK absorption modeling was to evaluate the impact of asciminib dose, different dose on the predicted magnitude of the asciminib PK change by elevated gastric pH. There was a lot so out of breath. But in this case, applicant already conducted clinical DDI study with the proton pump inhibitor, and they used lower dose of asciminib in the indication, which they did not see the clinically meaningful DDI, pH-dependent DDI effect. But because this type of interaction is more, the solubility change is more sensitive under the high dose condition, FDA issued an information request to the applicant and addressed the effect of different dose in terms of expected gastric pH increase effect on asciminib PK. So applicant submitted the two-stage in vitro dissolution study result and the PBPK modeling in response to this IR. So here's again model development, validation, and application workflow. Again, the relevant in vitro data as well as in vivo PK studies were used for model development and validation, focusing on the PBPK objective of assessing the impact of drug dose in terms of the effect of elevated gastric pH on the drug exposure. So here is the model application result. So PBPK addressed pH DDI potential of asciminib. So it well captured the observed not clinically meaningful impact of elevated gastric pH on asciminib PK parameters. And it predicted the Cmax and AUC difference due to the under the elevated gastric pH condition. And both at the 40 milligram and 200 milligram, the impact were not considered clinically meaningful. And also PBPK absorption modeling provided the mechanistic explanation. In this case, because of the high solubility in biosalt, the supersaturation actually overrides the expected pH effect, and thus resulting in the not clinically meaningful effect of acid reducing agent. So asciminib case summary, the applicant's PBPK absorption model was adequate for the purpose of evaluating the dose effect in this example. And it was used to support regulatory decision in conjunction with the two-stage in vitro dissolution data, which showed supersaturation evidence, provided supersaturation data and evidence, and also the clinical DDI study that was conducted using the lower dose of asciminib. So the regulatory impact, it provided support to the product labeling. Okay, so let me switch it to another group of example in terms of, in related with PBPK absorption modeling in support of the pediatric drug development program. So PBPK modeling with other modeling approaches has been used to support dose selection for pediatric drug development. So pediatric drug development and support by the modeling and simulation, it's a best amount, the very wide area. So I'm probably and definitely not covering all the aspect of the utility of the modeling in this area. And the case examples are very specific, sometimes very narrow focus area. But anyway, I wanted to share that the PBPK and other modeling has been supporting the pediatric drug development. And PBPK modeling can also support pediatric formulation development, which is more relevant for today's discussion. So the advantage of using PBPK model, as we all know, that it can provide mechanistic way to account for age-related differences in absorption, as well as other processes in the PK, the whole ADME. And also it provides a mechanistic way to bridge the formulation and bridging the observed relative bioavailability NBE that was conducted in adult population. Overall, providing support from adult to pediatric extrapolation in the pediatric drug development program. So the next case is with entrectinib, and the PBPK objective was to predict entrectinib PK in pediatric population where they don't have pediatric PK data under age 4, following administration of different formulation. So a little bit background here, why they, what was the motivation for PBPK modeling in this case. Entrectinib is also a low solubility drug with pH-dependent solubility, and this drug is mainly cleared by metabolism by CYP3A4. So during the drug development program, there were multiple formulations were developed, and initial formulation F1 shows very significant pH DDI effect, which could result in down the road food effect. So different formulations were developed subsequently. In this case, F2A and F06 are relevant for this discussion. So F06 was the to-be marketed formulation at the time being considered for pediatric use. And F2A and F06 were shown bioequivalent in adult population. So, but in the early pediatric clinical studies, only F1 was used. Therefore, the need for formulation bridging as well as predicting pediatric PK. So here's applicant pediatric PBPK modeling workflow. So they used 2 different platforms, and for pediatric model A, so adult PBPK model was developed for those different formulations. In this case, formulation difference was simulated or modeled rather based on empirical approach. So PAP permeability was varied dependent on the formulation. So which makes the extrapolation from adult to pediatric a daunting task, because it is hard to rationalize why we are extrapolating different formulation from adult to pediatric, by using the permeability difference. So due to the reason, model A was not further considered for pediatric PK prediction application. So model B used bottom-up, more mechanistic absorption modeling approach, and they extrapolated the formulation difference from adult to pediatric, and during the extrapolation, the age-related differences in this case, biosolic differences was used as a rationale, and also different type of CYP3 ontogeny was tested as sensitivity analysis. If you remember, this drug is mainly metabolized by CYP3A4. And then they used the developed pediatric model to predict the PK under age 4 for the very young children. So in this case, FDA concluded that both models were not acceptable for the proposed purpose of predicting PK under age 4. And this case is a good example highlighting the current challenges and the gaps that we may want to fill rather rapidly to expand the application of this tool. So the first one is mechanistically describing the absorption. As you can remember from the model A, the formulation difference was empirically described, and I mean, from adult to pediatric extrapolation, the age-related differences in GI physiology and what's the impact of the formulation, those should have been, linked or described, but it was hard because it was not described mechanistically. And the other one is also a related one, formulation difference not mechanistically described. And also, this is not just for the absorption modeling part, but clean farm application. If you remember that absorption modeling and the systemic model is combined to predict the outcome for clinical PK significance assessment. So tendency to overestimate the pediatric exposure. So they had the limited data under age 4 in the later phase of the program. And it turned out that the model was over predicting the pediatric exposure. So even with the alternative description of the ontogeny, so uncertainty was definitely both in the absorption and also the systemic disposition part. So again, this case is a good example highlighting the current challenges and uncertainties in the pediatric PK modeling, PBPK modeling, as well as the pediatric absorption modeling alike. So the other example is rivaroxaban. This case, just want to note that rivaroxaban pediatric PK program modeling and simulation were applied throughout the program. So not just PBPK, but population PK as well, providing support for dose selection for pediatric trials. And the example I'm talking about is very small piece. But since this is related with the age-appropriate formulation, I wanted to make a case of this. So in this case, PBPK modeling was used to support the use of the pediatric formulation, how they should be prepared to dose in younger children. So age-appropriate, from a little background, age-appropriate formulation, which was the granules for oral suspension, that was shown BE to the adult formulation, the II tablet formulation in adult. But there was some unexpected difference in the younger children. So on the left side of the plot, you can see it may be too small, but the black dot is the adult formulation, and the red is undiluted pediatric formulation, and the green is the diluted suspension. So all three formulations, or the way differently prepared pediatric formulation, were bioequivalent in adult, but they saw the delayed in absorption in younger children only. You see the red dot, decrease in Cmax and delayed Tmax. So PBPK model was used to explain providing mechanistic explanation based on the dissolution rate when the pediatric formulation prepared in two different ways. So rivaroxaban case summary, it provided mechanistic explanation for the differences in the pediatric formulation performance in younger children, and consequently body weight adjusted dose of the diluted pediatric formulation was used in subsequent pediatric trials. Okay, so I believe this is the last example in my presentation. This is the new area for potential opportunity for using the absorption PBPK modeling in clinical assessment. So recently published penebrutinib drug-drug interaction study with itraconazole in solution. They found an unexpected result with the itraconazole with penebrutinib. They expected increase in Cmax and AUC, and therefore to support fraction absorbed, fraction metabolized CYP3A4, but Cmax was rather decreased. It was lower, and even though AUC was increased, but the Tmax happened much later than it was expected. So in this case, PBPK model was in a retrospective way to support the author's hypothesis that this confounded DDI study result is based on the penebrutinib and the hydroxypropyl beta cyclodextrin present in itraconazole oral solution. So interaction between the investigational drug and the excipient in the index substrate drug that was used in the DDI study. So again, this example is based on the literature published case study. So the left side is the PK comparison with penebrutinib PK with and without itraconazole oral solution that contains the cyclodextrin. As you can see, the PK profile difference is somewhat unexpected. So they hypothesized that the presence of cyclodextrin that was that was in the itraconazole oral solution to increase the itraconazole solubility. Actually, penebrutinib showed very high complexation potential with cyclodextrin. So penebrutinib is also forming complexation with cyclodextrin or binding to cyclodextrin within when it was concomitantly administered with itraconazole solution. So cyclodextrin, while it increased the solubility of insoluble drugs, but it also affects the free concentration that's available for absorption. So that's why there should be a balance between the solubility and the permeability or absorption. But anyway, in this case, since penebrutinib has very strong complexation potential to cyclodextrin, in vitro studies supported that it actually increased the solubility in solution, but decreased the pF in the in vitro MDCK assay. So using this in vitro information, PBPK model was put together for retrospective analysis. So since this kind of availability describing the binding to excipient in the co-administered drug, it's not exactly the current capability. In this case, the PBPK model was used to predict what's the concentration of cyclodextrin in the GI tract. And using that cyclodextrin concentration to predict what's the permeability change, as you can see in the middle bottom, it affects the pF value. So translated into the changes in PF in the PBPK model, and they explained or captured the observed cyclodextrin and the phenylbrutinib interaction. In this case, it is actually cyclodextrin that's present in the itraconazole that's concomitantly administered with phenylbrutinib. But PBPK model was used to explain the underlying hypothesis in conjunction with the in vitro studies. So this definitely shows the potential of using the absorption PBPK modeling with further development to explain this kind of interaction. So this can be used to support addressing similar drug excipient interactions that may arise for other drug products, not just with itraconazole or other drug products or other excipient. Okay, so thank you so much for your attention, for listening to the case studies. So I want to conclude with overall summary that we acknowledge that the regulatory experience is being built in terms of the applying the oral absorption modeling to clinical pharmacology decision-making regulatory applications. So we wanted to share the current examples from a point of view, again, to provide insight into the potential opportunities present here, as well as kind of sharing our wish list for further development and improvement in this area to increase our competence when we review this kind of modeling. So we see that the current gaps may, largely, we summarized in the 3 different areas, which is very broad. So probably covering a lot of points that have been discussed in the past 2 days. The first one is the demonstrating the ability to proactively predict the drug formulation effect on in vivo drug PK. And the second one is the in vitro to in vivo extrapolation of the key absorption-related parameters. So from my personal view, this also includes the development and improvement of in vitro tools to be more predictive of in vivo PK behaviors or absorption behaviors. And also using the pediatric example, the understanding of age-related differences in GI absorption physiology, especially very young ages. This one not only applies to pediatric population, but also applies to other conditions, factors that may affect the absorption environment in the GI, like disease or use of drugs that can affect the absorption environment in the GI tree. So I hope that these were the useful discussion and case examples. And I want to acknowledge the PBPK team who provided all the case examples and the people who worked on these reviews. Thank you so much. And I think I overspent it.
So thank you, Miyang. But in the interest of time, we'll skip Q&A. Please feel free to ask her questions during lunchtime and during breaks. I'm going to ask our next speaker, Dr. Christopher Tannergren, to come over. And he's a principal scientist at AstraZeneca, and he's going to talk on prediction of regional/colon absorption and modified release drug product performance.
Thank you very much. Hello, everyone. Today, I have worked at AstraZeneca exactly 20 years. So it's an anniversary for me. Yeah. And when I started, I was told that you are going to focus on colon absorption and you are going to focus on absorption modeling. That's what I've done for 20 years. So I'm really happy to come here today to this fantastic workshop and present some recent work. And it's really special to me that I've been able to sort of combine the two. So going forward, I only need to focus on one area. It's a sort of modeling of extended release formulations. So let's start. I will give a quick background and introduction, and then I will start talk about the use of PBBM to predict colon absorption in humans. And then I will switch focus to PBBM regarding extended release formulations. And I start out with the Verinurad case study. And then the final part of the presentation will be around different ways to account for colon absorption when you predict in vivo performance of extended release formulations. And then I try to come up with some hopefully smart conclusions. So I think you heard when Xavier showed the results from the survey yesterday that the majority of applications for PBBM is on immediate release drug products. And I think it was approximately 20% of the applications were on extended release formulations. And I think we can all also agree that prediction of extended release formulations are more complex than for immediate release, because for one thing, you need to account for any regional differences in absorption and perhaps gut wall metabolism, etc. So, and especially colon absorption is a specific area of interest here. And it has also been identified as a sort of gap in our biopharmaceutics modeling and simulation capability. And I hope that everyone after this talk, if you don't already know it, understand that colon absorption is key for successful extended release product development, at least if you develop a profile longer than 3 to 5 hours. And actually, in my experience, is that it's not technical difficulties that sort of results in an unsuccessful ER development. It's actually most often limitation in colon absorption. And the reason for that is that the colon is a formidable barrier against absorption. The amount of water available for dissolution is really low, and we don't have any micelles there anymore that can help out. But perhaps the most important factor is that the surface area in the colon is so much lower compared to the small intestine. And on top of that, there are massive amount of bacteria in the colon that can degrade your compound. I have a recent experience within AstraZeneca, a candidate drug tested in our fecal slurry method, and everything was degraded within just a few minutes. It doesn't happen that often, but it can happen, and it can have massive impact. But thinking about modeling in this area, I think if we could come up with PBBMs that are appropriate at different stages of development to be sufficiently predictive, I think that would provide us with several benefits. So, for example, we could make really early assessments around probability of success when it comes to ER product development. And we could also reduce development time and costs by making smarter product design and hopefully also running fewer bioavailability studies. And ideally, of course, in the end, we would like to have sufficiently good models so we could actually apply the models for regulatory applications. So on this slide, I've tried to, it's a bit busy, but I've tried to sort of provide an overview of how we assess colon absorption and extended release drug product at different stages of development. And I'm quite happy to say that during lead optimization now, we actually have predictive in vivo predictive in vitro methods regarding permeability, and we also can utilize the dose solubility ratio to assess the impact of solubility and dissolution on colon absorption. And we also have colon stability assays to assess the degradation risk. And in preclinical settings, there are dog models available. There are porter dogs where you can dose directly to the colon, and I think there are also colonoscopy techniques that you can use. And moving into phase 1, there has been this possibility to run human regional absorption studies where you dose your drug directly to the colon in order to calculate the relative bioavailability in the colon. And today, then we have sort of made the decision that, yes, we can develop a ER drug product or not. And then, of course, later on, you get clinical data on your ER prototypes. But the sort of role of PBBM here has not been very clear. There are certainly cases when it has been applied, but it's not a clear strategy how to utilize PBBM throughout the development for ER drug products. I wanted to share with you the decision tree we have to share how we use the in vivo predictive in vitro methods to sort of assess the colon absorption limitation risks. And it's based on permeability, dose solubility ratio, and degradation. So more or less, if you have a high permeability compound, you have a dose solubility ratio around 30 to 50 milliliter. And I want to remind you that for a BCS class 1 compound, it's 250. So it's quite tough. And then, of course, if you have a stable compound, then the colon absorption limitation risk is low. But then the lower the permeability, the lower the solubility, the higher the risk will be. So moving into to talk about modeling, which we are supposed to do here. So we have done a recent work around evaluating how well we can predict colon absorption in humans using PBBM. And in this work, we established a new data set. It's 19 compounds. As you can see, it's a very wide range of permeability and solubility data in this data set. And all these compounds have clinical data when the compound has been dosed directly to the colon in humans. And as you can see, it's also quite a challenging data set with the relative bioavailability in the colon and the estimated fraction absorbed in the colon ranging between 3 to 100%. So, yeah. So we recognize the level of challenge here. So the modeling strategy, we went for a priori modeling approach. And the compounds we selected sort of allowed us to be sure that this would be a absorption focused evaluation. So on purpose we didn't include the compounds with a high risk for gut wall metabolism, etc. And the prediction parameters we evaluated was the AUC in the colon, the relative bioavailability in the colon, and the fraction absorbed in the colon. The last one was only done for solutions. And before we did the evaluation, we set 2 levels of the sort of prediction criteria. One as for highly accurate performance, and then we set an AAFE value less than 1.25 to sort of reflect a situation where the model performed so good so we could actually use it for sort of commercial product development applications. And then we set another level, which we called accurate, which was more or less a prediction within a 2-fold prediction error, more reflecting a model that is good enough to use in candidate selection and early product design. So on the right-hand side here, you can see the results. On the y-axis is the ratio between the predicted and the observed values. And on the x-axis is the effective permeability. And the vertical dotted line is the PF of metoprolol. So this means that on the right-hand side, you have high permeability compounds, and on the left-hand side, you have low permeability compounds. And as you can see, the predictions for the high permeability compounds were quite good, although we didn't achieve the sort of highly accurate criteria we set, but it met the sort of the accurate criteria. On the other side, you can see that for the low permeability compounds, the prediction performance is poor, and it's a bit interesting to see that GastroPlus under-predicted while the AstraZeneca software GISIM over-predicted, showing the differences between the softwares. I included this slide just to show the sort of difference between non-solutions and solution, more or less saying that there were no real difference in prediction performance between the different formulation types. I just want to say that all non-solutions were high permeability compounds. So that was around prediction of colon absorption. Now we will move into prediction of extended release formulations and the Verinurad case study. In contrast to lesinurad, Verinurad is a BCS class 1 compound. It has a pH-dependent solubility, and this means that it will also have a sort of pH-dependent release rate. And based on the decision tree I showed previously, Verinurad is a low colon absorption limitation risk compound. But you will see here on when it comes to the clinical data on the right-hand side, when we develop a phase 3 prototype and compare that with the phase 2 formulation, as you can see on comparing the dissolution profiles in pH 6.8, if anything, we actually expected somewhat higher exposure with the phase 3 prototype. But as you see in reality, the exposure with the phase 3 prototype was actually lower. And we wanted to understand why that was the case. So I did a traditional numerical deconvolution to get the in vivo release rate, the in vivo absorption profile. And that's the purple profile on the right-hand graph. And as you can see, in vivo, there was this almost 2-hour lag time. And then the in vivo release kicked off. And after 5 hours, absorption ceased due to colon absorption limitation. So in reality, we only had an absorption window of only 3 to 4 hours. And estimated fraction absorbed for the phase 3 prototype was only 40 to 50%. So we wanted to try something to go for a faster release profile and try to maximize the release in the small intestine. And to do that, we went for a combined in vitro dissolution and PBBM approach. It's a bit busy slide, but I will go from the left to the right. We saw when we estimated the in vivo release, we saw that the 6.8 release method didn't capture this initial lag time. But we also saw that with the pH shift method, as you can see here, then we capture the lag time and the in vivo release rate very good, except for the colon absorption limitation. So to account for that, we optimized the colon ASFs to match the in vivo release of the phase 3 prototype. And then we used that model to sort of evaluate how good it could predict other prototypes that we had. We had the IR tablet, and we had other extended release prototypes that we had tested previously. So we used that to validate the model. And as you can see, the resulting PBBM worked quite well. We had I can't really see, but I think the prediction error on Cmax was 8%, and on AUC it's 13%. And we could predict both immediate release formulations and ER formulations with different release profiles quite good. So we felt that, yeah, this model is good enough. So then we used this to come up with a newer, faster release profile. And we manufactured that and tested that in a BA study. And as you can see on the right-hand side, the observation that we made 6 months later exactly matched the prediction we did. So it went really well. And now comparing the fraction absorbed with the new formulation compared to the old one, we went from 45 to around 70%. So we were quite successful in this case. But of course, this raised the question: can this methodology have a sort of wider applicability, or was it just were we just lucky for Verinurad? But moving into that part, I just want to share that this model could actually have wider applications as well. So the model we have now is based on the pH shift dissolution, but if we then put in the 6.8 release profile, then we suddenly have the sort of worst case scenario from a PPI ARA DDI scenario. So that is a simulation we can do. And we can also use this to sort of try to simulate and predict the impact of the effect of alcohol in the presence in the gastric and the small intestinal content. And we did that by adding 21% alcohol to the gastric media for 2 hours, which is the highest concentration measured in the stomach and the small intestine after intake of 40% whiskey. And 2 hours is a longer time period than what was seen in vivo with this type of concentrations. And as you can see, there was no impact of alcohol, no dose dumping indicator. So final part of the presentation, and this is a work that I hope will be sort of accepted for publication soon. We have submitted it. And here we evaluated different approaches to account for colon absorption when you do prediction of extended release formulations. So in this case, we selected 5 model drugs, and we have in vivo data and in vitro release profiles for at least 3 variants for these model drugs. And in addition, we also had human regional absorption data. So we had data when the compound had been administered directly to the colon. And then we used this and tested 3 different scenarios. The first one was going for normal bottom-up modeling. And then the second one was then we took the observed plasma data after administration directly to the colon, and then adjusted the colon absorption scaling factors to adjust the colon. And then thirdly, we did the same, but more or less with a Verinnerad approach. We took the slowest variant and adjusted the colon absorption scaling factors, and then tried to predict the extended release formulations. So sharing some results for first metoprolol, which is a low colon absorption limitation risk compound. And on top you have the bottom-up approach. And for the low risk compound, the bottom-up approach looked quite good. I think especially for GI-SIM. For metoprolol, it's sort of a tendency for under prediction, and I think that is related to what we saw for when we did the colon absorption prediction, that in GastroPlus, metoprolol tended to act more as a low permeability compound, so it kind of agrees there. However, using the sort of colon absorption approach didn't really improve the prediction. If anything, it looks worse at the later time points. But the slowest ER approach resulted in quite good predictions of the extended release in vivo performance. And then for AZ2, which is a medium colon absorption limitation risk compound, then the bottom-up approach didn't look that good. The colon absorption approach didn't really add any value. But again, the slowest ER approach resulted in good predictions. And the same was observed for the high colon absorption limitation risk compound, AZ1. So trying to summarize the results, and here we also had 2 levels of prediction criteria. A prediction error of less than 20% we felt was a sort of highly predictive model. And if we had a prediction error between 20 and 50%, we felt that we said that was an accurate model. And if we look at the left-hand side on top for the a priori approach, it shows that this approach is accurate for low to medium colon absorption limitation risk compounds, but certainly not if you have a high risk. And as you imagine, based on the previous slides, the colon absorption approach didn't really add any value. But for the slowest ER approach, there was a significant improvement for all compounds, regardless of limitation risk. And as you can see, not for everyone, but we are quite close to reaching the 20% prediction error for all scenarios except for low permeability compounds. So to me, I think this is an opportunity to try to go for this approach. Trying to conclude what I've said, when it comes to prediction of colon absorption, PBBM can be used to predict regional and colon absorption in humans for high permeability drugs. And we can use this for candidate risk assessment and early product design and development. Low permeability drugs are poorly predicted. When it comes to prediction of in vivo performance of ER drug products, we can use the a priori approach in early product development and candidate risk assessment. And the slowest ER approach resulted in significantly improved prediction performance. And as I said, this is an opportunity to, it's not a guarantee, but I would certainly go for this and test it if it would work. And there is an opportunity to actually come up with a highly predictive model with this approach. And the exception is for permeability limited compound. And I think this is also an opportunity to streamline your BA studies. So instead of testing 3 different variants, you should go for the slowest one, generate in vivo data and make predictions based on that. And I think running a human regional absorption study where you dose the compound as a bolus to the colon doesn't really help PBBM development. And final conclusion is that we still need to work to improve the current colon models. And then before I stop, I will come back to this slide. And I think now we actually have handles to work with PBBM approaches when it comes to colon absorption predictions and ER predictions. And of course, it's a lot of persons that has been involved in this work. And I think particularly Harshad Yadhav, especially the last work, is part of his PhD. He's a Coloton PhD student. So if you want to discuss and learn more and collaborate, please reach out to him and me. We will be here during the rest of the meeting. Thank you very much.
Thanks, Christer. Again, in interest of time, we will not take any questions, but I'm sure he'll be happy to answer any questions offline. So now we have a rapid fire session of sorts where representatives of all the 6 agencies here today will speak 10 minutes about their experience with PBBM. So if the folks, as I call your name, if you could please make your way to the podium. Kim Rames, FDA, Louisa and Visa, Mary from MHRA, Evangelos, EMA, Shireni, Health Canada, and Hiroiki, PMDA. All right, so I guess we'll go in that order. So Kim, you are first up. And if I could please remind you 10 minutes. Thank you.
Thank you. So I know I'm standing in our way for lunch, so I'll go ahead and get started. Just want to thank the organizing committee for the invitation to speak on behalf of the FDA for this workshop. And this is going to be a high-level overview of PBBM in the current state. Disclaimer, this presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. So I'll get started. Patient-centric quality isn't new. It's something that we're continuing to work on and work through, and it's commonly seen in the number of guidances that we've currently approved and that we're still working on today. We hope to move forward. I often think of it, which comes first? We receive the submissions or the guidance goes out, but I think they work in tandem. So I think this is a partnership that's built between industry and the agency to provide accurate guidance, as well as get submissions that help to support moving forward. So the agency on a whole sees PBBM and pharmaceutical quality as a very valuable tool. If I had some more advanced technological experience, those three circles would rotate around the PBBM acceptance. We're looking at the totality of evidence. We've heard that in a number of presentations today that, we're not looking in a silo, everything is viewed together. It's knowledge base, and we're also assessing risk. So some of the key points that we've seen so far kind of leverage where that risk falls. So if we're looking at lower risk of the model, we may have a larger data set. There may be more knowledge available for there. It may be a low risk in setting that an acceptance criteria based off that model. But we also have that paradigm of going to things that are more of a higher risk, where there are more unknowns, there's a larger impact, and it has a large payoff. So if it's supporting a biowaiver or something where an in vivo study would have been needed. So I wanted to provide some high-level statistics of where we are as an agency today. So this is just a snapshot of just some key figures. So there's an estimate that we've had 50 or more submissions of PBBM or PBPK modeling simulations to support biopharmaceutics in both the ANDA, NDA space, as well as pre-development and IND submissions. Our current rate of improvement of acceptance or finding models acceptable is 48%. I think this is encouraging for the agency as well as industry that, we're moving forward with the continued knowledge space that we're building with the submissions that we receive. And currently, all PBPK modeling simulations to support biopharmaceutics are assessed as part of the regulatory submission. I think this is another way that we're moving forward, where there's active engagement with models that are submitted. They're not just sitting there and, not being evaluated or being looked at. So I think we're definitely moving in the right direction and supporting this field. A number of my colleagues have already spoken from the agency today on a number of the different cases that we've received using PBBM. So I'll just highlight, we've seen them in cases of dissolution method acceptance, patient-centric quality specifications for critical material attributes and critical process parameters, as well in SUPAC and risk assessment spaces. And this is kind of a pie chart to see how the distribution falls. Where are we seeing the most submissions in relation to PBBM? As you can see, there's been an uptick in the SUPAC changes. And I think we can, say that the guidance coming out in 2020 may have led to this uptake. I think there was more confidence and assurance, that the agency is evaluating these to support quality changes. And we were able to demonstrate that we could use them in those situations. One other thing, it looks like risk assessment went down. I'm not sure if that's really an anomaly or it was also a direct reference to the guidance, because I think now with the guidance, there was a clear understanding of what your model will be used for. So things have gotten more specific in their application, and those defining of those areas may have shifted some things that may have just been a risk-based to a specific something that they could help during the life cycle management of a drug product. Some other things that we've noted, since the publication of the guidance, we've received many dedicated modeling reports that provides ease of some review by our regulators, which is also often very helpful. We have a clearly defined model purpose, and that speaks to what I showed in the last slide. We're clearly able to see what's being articulated for the reason for the modeling, and that helps us in the evaluation to see if it's acceptable or if the area that's being evaluated for is applicable. There's scientifically justified selection of dissolution models. We've had a number of great presentations in this workshop, and I think the increased understanding of when to use certain dissolution models and what benefits there are has really helped in us having those justifications included in our packages. And we also have reliable model supporting files. I think some of the previous FDA presenters have noted that, we're rerunning these models. We need the data to support that for us to be able to accurately assess. And we've seen that this is now commonplace, and we're getting these data submissions for us to review. So we started with the good points. So what are areas where we've seen some deficiencies, areas where improvement is still needed? I kind of lump these into groups. So some of the things that we've seen are inadequate model development, inappropriate model validation. So with the model development, some of the optimizations may not have been justified, where we were able to clearly understand and define what they were used for. For the model validation, there's still some uncertainty in what your prediction error is, what is acceptable, what would be the validated model. And also we've seen some drug-specific anomalies. So these are kind of things where it's unknown. Like we've seen things where there's unexpected trends in dissolution that aren't justly discussed or understood. And we find that as a gap in our understanding and being able to clearly evaluate the PBBM submissions. So we acknowledge that there are challenges, and we hope activities such as this workshop provide additional clarity. But I want to say this is a shared challenge, that we're in this together and we're working towards these. So understanding the sparse in vivo data available for model verification. I know there have been some breakout sections on that, and we're open to discussions on how we can move forward in that space. The knowledge in the GI physiology continues to expand. So, scientifically, when more information is known and we can build that in and have a better understanding, that also helps for our assessment. We understand the constraints of model validation. We know there's different acceptance criteria, and there's not a level of clarity, or more clarity is needed in that area. And that's also something we're working on. And then we know regulatory harmonization doesn't happen overnight. So this is a process. We wish we had a switch that we could flip and everybody be on the same page and we could move smoothly, but we understand this is a process, and it is something that we're working towards. I think it's saying they want me to be done. So I'll conclude. The future state, what are we looking forward moving forward? So in vitro tests and physiologically based biopharmaceutics modeling that provides predictive insight into in vivo performance, and that this ensures high quality drug products that maintain safety and efficacy throughout the product lifecycle. And we hope that with the predictive dissolution and modeling, the impact of critical bioavailability attributes on in vivo performance can be qualitatively assessed. And this would provide scientific and risk-based knowledge to support patient-centric quality standards. So I want to thank everyone for their time, and I'll turn it over to the next speaker.
One back here. Good morning. I will present on the applications of PBBM in regulatory submissions under the perspective of Anvisa, the Brazilian health regulatory agency. Starting from the disclaimer, this presentation reflects my own views and should not be construed to represent Anvisa views or policies. Briefly, in the 10 minutes, I intend to briefly cover the potential applications of PBBM on drug development and regulatory decision-making, highlight some PBBM cases that were submitted and assessed by Anvisa, and present our perspectives on PBBM regulatory scenario initiatives. So at this point, I don't want to be repetitive, but we see a potential applications for PBBM on drug development with a unique valuable value for being enabler for quality by design and MIDD principles in corporations. So to gain enhanced knowledge about quality attributes that may affect absorption and decay, to optimize formulation and dissolution development strategies, and to optimize biopharmaceuticals assessment. And this application on drug development has an interaction with potential regulatory submissions. And for the regulatory decision-making, we always have to balance the PBBM impact with the risk of the proposal and the model assessment. So here I listed some of the applications we have already discussed a lot during this workshop. And here I present some statistics of cases that were formally submitted to Anvisa between 2020 and 2022. So we had received 6 cases in these 2 years. They were directly to Therapeutic Equivalence Department and also for the Office of Pharmaceutical Quality. They were submitted by both national, Brazilian-based, and the multinational companies, and for new molecular entities, and also for generic drugs. Here there are some more details about those six cases. We have the first 2 cases submitted for new molecular entities for supporting CMC post-approval changes. 1 case succeeded and the other were not approved. Both were a strategy for submitting PBBM in a case that dissolution with QC method failed in F2 tests and also in other statistics comparison. The first case, the company provided a rationale for building an IVABC for supporting a bio-waiver because for those CMC post-approval change, a BE study were required, but they have a lot of uncertainties on input parameters, and the company were not able to provide the correlation to validate the correlation proposed. So the model were denied, and the BE studies on both fed and fasted conditions were required. The case 2 company is a case of CMC post-approval change for which BE were not required. Only comparison of the solution profile would enable the approval, but it failed on F2, also on other statistical approaches. So along with root cause analysis of the failure, company proposed a PBBM VBE approach for this IR-BCS4 drug. After dealing with the model uncertainties, this case were approved. Case 3 and 4 came both from generic industries with the intent to bio-waiver different strength that the one that was studied in a BE. Formulations on both cases were proportional, but they failed on F2 submissions. And these 2 cases were unsuccessful. Case 3, company didn't have enough knowledge on the design space of the product to guarantee that the failure on the solution were not due to poor manufacturability. So the case were denied and the application were not approved. In the case 4, company tried to go with a model for providing PBBM and VBE, but also conducted the BE study, and while we were assessing the case, they withdrew the submission because the real study had another result. And case 5 and 6 came both from new molecular entities for dissolution specifications. Case 5, company saw out of specification results and proposed a PBBM VBE to approve the batches and also to change the QC specification. It is a IR formulation BCS1, and they this evidence with other evidence were able to approve this application. And on case 6 is an interesting case where the company submitted a very robust model that were performed under an MITG drug development. They provide a PBBM with a safe space and PK/PD relationship to justify the clinical relevance of the dissolution specifications. But in this case, for the agency at this time, we consider this model informative because the dissolution specifications could be set based on the discriminative capacity of the QC method. And here, moving for our perspectives and the initiatives. We see a great value in collaborations like this one for us to discuss best scientific practices and also possibilities of regulatory convergence and also reliance pathways. The timing of this collaboration was quite good for us because we started to receive the first case in 2020. For dealing with this, we have created at Anvisa working group with collaboration of academia. And in 2022-23, we had put more focus on discussion of the cases and also on training. And from this year forward, we want to expand our knowledge on other applications of PBBM, like mentioned here, for assessing food effects on BE, GGI impact, pH dependence on BE, and also local acting effect. We also should having more discussion with our local industry to understand how they are using PBBM in drug development, and also move forward for having assessment workflows established. So nowadays, PBBM acceptance for review by Anvisa is discussed on a case-by-case. We stimulate early interactions with the agents. We have a formulary questionnaire that can be used, and also meets can be requested with both departments I mentioned. We are working on alignments and amendments needed on our current legislation to create a base, both from the biopharmaceutical side and also to engage more on MIDD policies. We should continue working on actions to multiply the knowledge sharing and to institutionalize the discussions that Anvisa having further involvement of other offices. And we need to have biopharmaceuticals risk assessments and frameworks. And I believe that some good propositions will come after this workshop. Yes, with that, I leave my acknowledgements for the reviewers of Anvisa, for the academics that collaborate with us, and also for the industries from IQ Consortium for providing cases for our discussions, for the other regulatory agents for being available for discuss, and also for the software providers for providing us training. With that, I pass the words for the next speaker.
Thank you. I will be very brief because partly what I'm going to present has already been covered, and partly because MHRA's experience in PBBM is relatively limited. However, we have experience with PBBK, so this won't be a problem. And before you submit, you can let us know in advance, because potentially it may need a collaboration between quality assessor and clinical pharmacology assessors, but we don't envisage any problem in this. So we consider PBBM a very useful tool for product development of both innovative and generic drug products. And as we saw today and the previous days, it has been used for establishing dissolution safe space or the drug substance particle size distribution specification. here, we believe that at least when for the submissions to the agency, we would like to see the workflow and how it has been followed. Here are some take-home messages that I also covered yesterday. I will focus in some others, like the fact that we would like high-quality experimental data should be available for the model, or that any assumption should be defined and justified. Also, okay, this is a topic of discussion, but it is recommended to use intravenous data or oral solution data during initial model development to appropriately derive the disposition parameters and justify the methods and the tools you use. Also, it is advised that still another topic of discussion about having a non-B batch. And something else that we, by assessing together with the other regulators, the cases that were presented, we think that the acceptance criteria for judging predicted versus observed data during validation should be pre-specified, because sometimes we felt a bit uncomfortable with this, that the predicted, if it is twofold within the observed, this is okay. We think that we would prefer to stick to the narrower acceptance criteria for bioequivalence. Yes, this is what I wanted to cover from MHRA perspective. Thank you.
So, good afternoon from my side. I'm Evangelos from the European Medicines Agency, and I will be conveying the views and the experience of the EMA, some high-level points around the topics that we've been discussing the last 3 days. Okay, the standard disclaimer: these are personal views, of course. I won't represent the official EMA views. Okay, just to start with, I suppose you're all in this room aware of the guideline that we have at EMA. It's in force since 2019. I'm afraid the figures that you see in these graphs are a bit outdated. They come from a rather old publication, but nevertheless, it shows that the use, although there is modeling, it is used in lots of submission. It concerns other types of modeling. PBPK has been rather limited, and PBBM within PBPK, it's even less. Nevertheless, I can confirm, even though it was not possible to get the accurate, more up-to-date data, that these figures have increased over the years, and also the trend that you see in the other graph of number of submissions over the years continues to increase in EMA submissions. But in absolute numbers, it's still low, and therefore the experience of the remains relatively low. Okay, so generally what we ask when we receive applications, either as scientific advice, but most importantly in a market authorization, the most important thing is that we have a proper qualification of the platform for the intended use. You've heard that many times during these days, but it's a very, very important thing. And of course, as we move from low to medium to high impact models, then that's the requirement, let's say, increase. And we speak about numerical and computational verification of the platform. We would like to see clarity on the assumption, but also on their impact. We would like to see the prediction performance of the model to be well discussed, well documented. Mary mentioned some limits about the prediction errors. I mean, these are issues that often come up during the assessment of these models. And of course, the sensitivity analysis for key parameters are the most common things that we look for. This graph, again, it's not a new scheme. I'm pretty sure you have seen and you are familiar with that. Let's say we classify the models depending on their impact, and by impact, we mean the impact they have on regulatory decisions. From discussions with some of you yesterday, we understand that this is not necessarily the same from an industry perspective, but this is, and it will be important in the applications that these aspects are also discussed somehow, because we speak about global development and global manufacturing chains and so on. So obviously, as we move from low to medium to high impact, the level of documentation is increasing, and the scrutiny, the assessment scrutiny that goes into the models is increasing. Common deficiencies, again, this has been covered also by previous colleagues here. Context of use, sometimes it is unclear. We face the same issue even with the case studies that we have been reviewing. Some of them, at least, we have been reviewing in this project. Input parameters, their uncertainty should be sufficiently discussed. Of course, model verification needs to be very well described and performed. The prediction performance I mentioned previously should be supported by appropriate data. And I would agree with Mary that we would like to see that being pre-specified ranges. Yes, also the clinical relevance of any misprediction. We often have, over predictions, under predictions. We would like to see that discussed at the right level. Very often the inputs and from the dissolution method and in vitro testing, the methods are not well described, the development of the methods and so on. And of course, we need clarity about the different types of formulations, the batches, number of batches, size of batches, and so on in the that feed the models. Now, I don't think we've touched that, but there was this question in some discussions I had with you during these days and in the preparation of the comment. So where should the information be located in the dossier? And of course, it is a multidisciplinary area. So I would say that the drive, it depends on the driver. If the model is there to justify specifications, we would expect to see that in module 3. Of course, if it's elsewhere, it won't be a deficiency, but it's more logical that it is there. If it is to, I don't know, replace a study, probably module 5 is the right place. In any case, we would like to see meaningful summaries in module 2 as well, particularly for high impact models. And again, regardless of where it sits in the dossier, you can use cross-reference to avoid duplications, to avoid repetition, and facilitate the reviewers. We would like the actual reports that go into the dossier to be detailed and as elaborate as possible. Do not assume that your assumptions, the starting point, the actual intention, or the background, which usually is very rich in your organizations, is known to the reviewers. So please make it as explicit as possible. Okay, I would like to also take a couple of minutes to highlight the fact that the topic being a multidisciplinary, it sits at least on 3 legs: the PK leg, a modeling leg, but also a quality aspect leg. And we had recently a big reorganization of our working parties at EMA, Now, the pharmacokinetics, the former pharmacokinetics working party, our modeling and simulation working party, and our biostatistics working party all sit under the roof of the methodology working party. And this is something that would hopefully improve the interaction among those aspects. And of course, we would have the quality working party, which has also gone through a similar reorganization. And why I'm highlighting this is because I found very interesting that in the work plan of the methodology working party, you will see some points which I think worth highlighting. So, for example, it is in this work plan to develop a Q&A on reporting and qualification of PBPK models, another Q&A on food effect assessment and drug interactions in the GI tract, development of a concept paper on model-informed bioequivalence. I think these are all very much relevant to what we discuss. But also on a kind of non-guidance document, there will be effort and a project to identify applications of modeling and simulation that for some key aspects of regulatory submissions, and then also work on developing and adapting standards for creating a framework around those things. And of course, workshops like the current one, or shall I say, the overall project that we led to this workshop has been acknowledged, has been included in that work plan, and maybe future initiatives and collaborations and projects can be further detailed in the future. I think, that takes me to some take-home messages. Most of this has been mentioned already. So, of course, it is acknowledged that PBBM without modeling is a useful tool in building a strong scientific rationale. And of course, we know it's widely used to inform development. Definitely, it's subject to assumptions and uncertainties. However, it is accepted that it can be used to inform regulatory decision making. Of course, as I mentioned, depending on the impact on the decision making, more scrutiny, the level of scrutiny is adjusted and depends on that impact. It is a multidisciplinary issue. This is acknowledged, and I mentioned about the interactions and the new ways of working at EMA. And as again mentioned yesterday, the early dialogue with regulators is recommended. Scientific advice would be the right way to go. And I would like also to highlight what Flora mentioned before. It may went unnoticed, although I doubt that anything that Flora says goes unnoticed. That the parallel scientific advice that we have this framework with FDA. I think this is an excellent topic that can fit into that framework, and I would invite you all to take advantage of that. And with that, I would like to close my presentation, acknowledging a multidisciplinary team that has been put together, and the trigger for that was this project and the workshop that we are attending. I'd like to thank all of them. Some of them are here these days, and these are the colleagues that have been reviewing these case studies and helped me put together this presentation. And with that, I would like to close and pass the floor to next speaker, Rainy.
Thank you. So I'll be presenting some perspectives from Health Canada on PBBM and regulatory submissions. Just the usual disclaimer that this presents my views and not that of Health Canada. I think we're all aware that pharmacometric approaches are evolving tools and they're used throughout drug product development. But as regulators at Health Canada, we hardly see it at that stage. We tend to see it when it comes in to gain regulatory approval or during drug product lifecycle management. In 2021, Health Canada published a very high-level policy statement. And the main takeaways from that was just to inform sponsors of the principles to consider when filing drug submissions or clinical trial applications that use these approaches. The main takeaways were that the quality of the data and the analysis are important. The strengths and limitations associated with their use should be described, and the feasibility of alternative conventional approaches should be discussed. Health Canada is also participating on ICH M15, which is the general principles of model-informed drug development. Submissions that we receive are increasingly include evolving methodologies, such as biopharmaceutics modeling. And these are submissions that are submitted to the Bureau of Pharmaceutical Sciences, or BPS. So in response to this, in September 2022, BPS implemented an organizational change by creating a pilot team, DBE3, to respond to the increase in submissions we're seeing that involve biopharmaceutics modeling. This was aimed at providing dedicated resources for scientific issues that, other than modeling alone, also impact other areas of complex products and evolving methods. But it provides centralized expertise for biopharmaceutics review within BPS, and that's co-located with the divisions of the quality review areas. Other modeling, such as POP-PK or PBPK or PKPD modeling, is reviewed by our pharmacometrics team, which is in a separate bureau, the Bureau of Policy Science and International Programs. I think we've heard a lot over the last couple of days about regulatory applications in biopharmaceutics modeling. And at Health Canada, this is not very different in what we see. We see bridging of formulations during the clinical program, additional strengths when bioequivalence for one strength is supported by clinical data, but maybe does not meet the F2 criterion for proportionality, changes to manufacturing process of site, or a scale up of batches when it exceeds greater than 10-fold, and therefore we'd normally require biopharmaceutics data, such as a comparative bioavailability study. We've also received studies and modeling studies to support biopredictive dissolution methods for appropriate in vitro dissolution conditions for extended release formulations or low solubility products. We've also seen submissions for in vivo relevance of dissolution methods for complex drug products, such as long-acting injectables. Another wide area of application is safe space design for manufacturing controls. That includes justification of drug or product specifications and their acceptance criteria. So this gives a snapshot of what's happened since DBE3 started. So before that, reviews for biopharmaceutics modeling was handled by the two divisions that we have for biopharmaceutics evaluation, DBE1 and DBE2. Since the start of DBE3, over a 12-month period from September 2022 to August 2023, we've seen a twofold increase in submissions that have included PBBMs and IVIVCs. So these numbers reflect both mechanistic IVIVCs, sometimes for long-acting injectables, the traditional kind of IVIVCs, and as well as that, PBBMs. This also reflects the growing applications of PBBM, and I believe maybe an understanding from industry that our regulatory preparedness is improved. We've also tried to engage externally, partly in workshops like this, but also in pre-submission meetings and in industry association meetings. And we've had Arthur present at a local symposium for CSPS in Toronto in May. So DBE3 has tried to streamline the review process. And just to give you an overview, when a submission comes in, it's screened by a regulatory project manager and assigned to the different review streams. The review streams then start off by having a kickoff meeting just to identify major deficiencies. And at that stage, either a preliminary information request is issued, or if a major deficiency is identified, the review may stop and you might receive a negative decision. Progress meetings are then held throughout the review of the process. And usually there are 2 progress meetings that are held for a new drug submission. In between that, we may schedule discussions or consultations with other teams, and this involves the quality, pharmacometrics, and clinical review teams. And this is to get an idea of what they're doing on their end and if we need input from our end. The direct briefing meeting is then held to finalize a recommendation for the submission. Since we don't have PBBM guidance, evaluations are considered case by case with consideration for the context of use and available guidance such as the FDA draft guidance that was issued in 2020. For filing within ECTD, we usually look at the summary that's included in 2.7.1 when the RPM or regulatory project manager is screening the submission. So it's really important to have a good summary in there. We expect the reports to be included in Module 5 and 5.3.1.3, which is the IVIVC segment, with a file tag for PBBM reports. Right now, when we get in submissions, this varies across the board, and we do see reports within the quality modules as well as the M modules. Review template has been discussed yesterday. It was brought to my attention that we're the only ones who have a review template for PBBMs and IVIVCs, but I think we can all appreciate that our resources are limited, and this actually facilitates our review. So we tend to provide this to sponsors when it's screened in, for sponsors to fill it out to help us complete our review in a timely manner. Maybe as we move forward and things are more standardized and our reports across regulatory agencies are all in one place, and maybe this will improve and we would move to a more standardized format where we may not review this or need this. Another thing that's planned is a revision to the abbreviated new drug submission checklist to include an identifying box for modeling data, which currently we don't have. Concerns noted for PBBMs and submissions are similar to what the other regulators have described. Model objectives are sometimes not very clearly defined. Input parameter values are not adequately justified. Uncertainty in parameter estimates are not adequately addressed. Dissolution data input methods may not be discussed or justified adequately. And sometimes limitations that are noticed during PBBM development, but they're not actually gone through to providing refinements. It's just noted and the sponsor moves on. For model validation, data sets sometimes are not appropriate or they're inadequate, and validation criteria are not stated a priori. For model validation, application variability sometimes is not mechanistic or adequately incorporated in the virtual trial populations. And I think this has been a topic of discussion throughout this workshop. Model risk and feasibility of alternative approaches also is not adequately discussed in many reports I've seen. Oops, sorry. So take-home messages. I think a carefully considered modeling strategy and a well-organized, detailed PBBM report facilitates evaluation. In this regard, the checklist that's on the website for this workshop developed by IQ could be a really useful tool. Continued interaction and collaboration with other regulators, industry, and academia is also required to further regulatory applications of PBBM, especially within Canada itself. So in recognition of that, we're planning to continue with a virtual seminar series. And also in collaboration with CSPS, we're hoping to plan a PBBM workshop in Ottawa in fall of 2024. And I'd like to acknowledge Arthur Kumu, who's with the DB3 review team and is attending this workshop, and my director, Chris Rose. Thank you all for your attention.
Hello, everyone. Today, I'd like to talk about the status of model and simulation applications in Japan and the experience in reviewing drug application, which was proposed the acceptance criteria based on PBBM in Japan. The number of model and simulation applications submitted to the PMDA is steadily growing. I'll show you the trends of PBPK model analysis in new drug applications approved from Japanese fiscal year for JFY 2014 to 2020. PBPK modeling was performed for about 10% until JFY 2017. Then PBPK modeling has gained momentum since JFY 2018, increasing to about 25% of new drug applications. As shown in the previous slide, the use of PBPK model analysis in new drug applications is increasing in Japan. But the number of new drug applications that attempt to assure quality, such as setting acceptance criteria using PBBM, is still limited. So we don't have much experience in reviewing drug application using PBBM for quality assurance. The PMDA launched the Modeling and Simulation Project Team, or M&SPT, in April 2016. The purpose of launching the M&SPT is to address the increasing number of model and simulation submissions in recent years and to strengthen the framework for using and sharing knowledge and experience with M&S. The M&SPT collaborates with reviewer of clinical pharmacology, biostatistics, clinical medicine, and quality, and functions as the framework to enable the discussion beyond experience and therapeutic areas. Shinichi Kijima leads M&SPT, and I'm joining the team for addressing the PBBM application as advisor from quality reviewer. The M&SPT work with various project teams and reviewers depending on the issue. M&SPT supports evaluating PBPK modeling, concentration QTC modeling, pediatric extrapolation, acceptance criteria supported by PBBM, and so on. When acceptance criteria using PBBM were proposed in drug applications, quality reviewer worked with M&SPT to review. This slide shows the number of support cases of M&SPT for assessing M&S applications in clinical trial, consultation, and new drug applications. The right pie chart shows the number of cases by the purpose of the model used. As you can see from this graph, the most common object of model and simulation is dose finding or setting, especially for phase 3 trials and new drug applications. Since the number of quality assurance using PBBM is limited, those cases are included in the other category. This slide shows the role of review team and the M&SPT in joint evaluation. Firstly, the content of use are defined for each question of interest to outline how the model will be used to inform the question. In this step, the review team often has to consider the position of clinical data package and M&S, Therefore, it is necessary to come to understanding about COU between review team and M&SPT. Next step is evaluating adequacy of modeling strategy, verification and validation of the model, and simulation scenarios. Since this step highly specialized regarding model and simulation, the review team requests the M&SPT to support. M&SPT supports assessment of M&S applications for individual products based on the verification and validation activities, history for the constructions of the models, and so on. In the credibility assessment of M&S applications, the M&SPT primarily assesses validity and result of the verification and validation plan, and the review team reaches the final decision of the acceptability of M&S. Next, I will talk about some of the considerations in the review of the drug application which were set acceptance criteria based on PBBM. For example, we evaluate the following point: the model's contribution in assuring the quality of the product, model credibility according to its purpose, adequacy of simulation scenarios using models including parameter-sensitive analysis, the justification of acceptance criteria submitted for the product. It may be necessary to consider a reasonable range of acceptance criteria, including model uncertainties. Okay. In closing my presentation, I would like to go over the key points. In Japan, the number of drug applications that attempt to set acceptance criteria using PBBM is still limited. The PMDA launched the M&SPT, and when acceptance criteria using PBBM was proposed in drug applications, quality reviewer worked with the M&SPT to review. In the review, the model credibility is the model's contribution in assuring the quality of the product, and appropriate acceptance criteria are also considered important. That's all. Thank you for your time and cooperation throughout this presentation.
So I'd like to thank Kim, Luisa, Mary, Sharini, Evangelos, and Hiroyuki for their presentations. Please feel free to talk to them during lunch.